Okay, well, good morning, everyone. We're going to start the session without further ado, and the first speaker is going to be Margaret Byrne, who's director of a new structure which has grown much bigger in WA. She's executive director of the Biodiversity and Conservation.
Science.
Yes,
don't forget the science bit. Thank you, Renee. The science bit's the important bit. Thank you. It's a pleasure to be here, and thank you to the organising committee for coming back to celebrate the 20 years since we first brought the group together to talk about eucalypt genetics. So what I thought I'd do this morning is just do a very quick run through a whole range of different things that we've used genetic markers for to understand eucalypts and their diversity and how we can manage them and how we can manage them into the future. And you'll hear, so that's just with a few examples from the work that I've done, but of course the rest of the day we'll give you a whole range of other examples in more detail that other people are working on. So in 2008 I pulled together a chapter for the plant genome series on eucalypts and went back and looked at all the diversity statistics from all the range of different markers that have been used and looked at them. So what I thought this time is I'd just update it. There were only a few microsatellite studies at that time. Just update it with the diversity stats for microsatellite studies, because in the early days, back 20 years ago, that was in terms of molecular markers, that was a lot of the kind of things that we were doing, and particularly from even before that with all the allozyme studies. So this is just a summary for widespread, regional and localised species, following that summary of distributions that was worked out really early on. And you can see that eucalypts are very diverse. There's a lot of diversity. Now, of course, everybody who's done anything in terms of reference genomes knows that the heterozygosity is a bit of a problem, but in terms of the species, persisting and adapting and managing through in the landscape, heterozygosity is pretty important. And you can see that widespread and regional ones, you maintain pretty good diversity. If you get localised species, you can get a bit of a drop in diversity. How's that diversity structured? So a lot of the studies looked at not just diversity, but how is that structured across the landscape. And for 54, so this includes all markers now, 54 looking at patterns of FST, which is a general measure used for population differentiation. And it's generally reasonably low. So for widespread species, about 0.08 as an average of 21 species, but in really semi-continuous distributions, forest-type environments, it's down at 0.05 and some species down as low as 0.02 and 0.03. So really got not a lot of structure across widespread species, get a bit more in regionally distributed species, and then of course quite a bit more in localised species, because they generally are pretty patchily distributed in the landscape. So that's just kind of a summary of the background of the diversity and differentiation across a range of species of eucalypts work that we've used genetic markers to look at across the board. We're now into genomics, and I didn't pull together all there's a handful of genomic studies now done across the species distributions, and sometimes done in the same type of markers and some different. So I didn't try and pull the stats together for the genomics data, but just wanted to give you two comparisons. This one's a particularly good one because it's using the exact same populations. So we've got a microsatellite study of 28 populations across the range of Marri, which is a main forest tree in Western Australia, and then a genomic study across the same individuals and the same populations. So you can see in microsatellites that we've got a classic structure line of just isolation by distance from the north to the south, no major differentiation. The FST value is 0.03. And then when we've done more work, this is with Colin Arends and Paul Reimer, and looked at the same populations, we get slightly higher FST of 0.05 and detecting more fine cell structure. And the one that we think here is this structure plot here, where we've essentially got a southern group, a central group, a northern group, and then a far northern group. And you can see that the individuals all cluster quite tightly into the populations, which if we'd have done a PCA with a microsatellite, you just usually get individuals all overlapping. So we detect, with the power of genomics, we're able to detect much more fine-scale genetic structure across widespread species. And here's the latest one. Hopefully you saw Zhao's poster last night. So this is Jarrah, which is the other main forest tree distributed across the southwest. And this is an old RFLP dataset where we found no structure across the forest. And the red is the northern forest and green is the southern forest. Foresters differentiate between the northern forest and the southern jarrah forest. We found no genetic structure with RFLP loci, but with genomics we've found structure again into the north, the centre and the south. And interestingly, the coastal plain come out. Of course, we've got Gillican rock, which is a known outlier out here, just a few plants around the granite outcrop. But this differentiation here with the ones on the coastal plain versus the scarp, so if you're on Perth, you've got a coastal plain and then an uplift about 300 metres onto the scarp. We did detect that with a chloroplast study where we found the coastal plain was a different clade to the plateau clade. So interestingly, that the genomics data is now picking up that differentiation between the plants on the coastal plain and the ones up on the scarp. And when we've done a rough molecular clock dating of that, that's dated at, it correlates with the data with the uplift of the Darling Plateau, which was at the end of the Pliocene 2.8 million years ago. So, what else have we used genetic markers for? So a big lot of studies have used it to understand the mating system of eucalypts. They've got generally a mixed mating system with dichotomous pollination, generally mass flowering with small flowers, although you do get some really large flowers, particularly the red bird pollinated flowers, flowering species. And so a lot of studies have looked at outcrossing. So originally in 2008, when I looked at the average, so we had an average of 0.74 in terms of outcrossing across 23 species with a range of 0.5 to 0.96, and I've just updated it. We've now got 30 species that have got mating system studies, and we've got very similar average, and that followed on from the work that Brad did in an earlier publication, which I think had about 17 species, and it was 0.73. So the mating system, the understanding mating system is staying pretty constant despite the number of studies and the different marker systems that we're using. This dropped down to 0.08 is one study. That's Eucalypt sapsita, which is a clonal species. We've got clonal patches, and so it's actually got a lot of selfing in it, but it's able to tolerate actually quite high levels of selfing. Seed dispersal is generally pretty restricted, and pollen to seed ratios where they've been calculated are usually pretty high, so most of the gene flow is occurring through pollen dispersal. So studies have also looked at pollen dispersal, particularly with paternity assignment, with the greater power of microsatellite markers, and it can be really quite extensive with long distance dispersal and a fat-tailed dispersal kernel. Prior to these kind of studies, it was generally thought to be relatively a leptokerpic distribution with a very thin dispersal kernel. So in fragmented landscapes, here's just a few studies that showing that it can be quite extensive over 2 to 3 kilometres, and those figures are really just constrained by the fragmented systems that we work in and the distance over which populations are distant from each other, and it could actually be quite a lot further than that. And that was a bit different from previous studies, which had often been done by looking at hybrids, so Knighton's ovada hybrids, and looking at pollen dispersal into those and looking through morphological traits to detect it in the seedlings, where in continuous forest systems, pollen dispersal is usually quite constrained and does follow that classic leptokerpic distribution. But when you get into fragmented landscapes, you can get quite extensive pollen dispersal. Now, of course, eucalypts are known also to have clonality. They've got a lot of them have got an underground lignituber, and so are multi-stemmed, arising from that lignituber. But what we've been able to show with molecular markers is just the size and extent of those clonal patches. Some of them can be quite extensive, so often 20 to 50 metres, not uncommon, and you might have a 50-metre diameter clone. You might have up to 20 to 30 different stems in that, and you can walk between them. Eucalyptus violaceus that Rob mentioned yesterday is over 3,000 square metres, and there's actually a car park in the middle of it in that one. And age, where they've been dated up to 6,000 years old. And some of them can have just basically exist in clonal patches. So Eucalyptus sapsita, just giving you the distribution here. You probably can't read it. There's whole series of patches just in here, and one over there. There's 14 populations of these clusters where it occurs across a ridge in agricultural land, and each one of those patches is a single clone except one. This one patch has got 2 clones in it, so it only exists as clonal patches. And other species can have a mix of clonal and non-clonal populations, such as Eucalyptus caprea. They're both 2 kind of rare species. We talked about hybridisation yesterday, and of course you've got hybridisation at all levels, and there's a lot of need to understand the articulate kind of evolution and the role that hybridisation has played in that in eucalypts, and it looks quite extensive. But it's also important at a management level and for conservation of species. So a lot of actual species, or quite a number of species, have actually been recognised taxonomically as eucalypt species, turn out to actually be hybrids, and often are able to persist because they've become clonal and they're just a clonal patch, multi-stemmed and often single individuals. So here's just an example of Eucalyptus bennettii. It was listed as critically endangered on our declared rare flora list, which of course means we were supposed to put a whole lot of management resources into protecting it. It's known from two populations. There's a report of a third one, but they weren't able to find it when they went looking for it. And it was believed to be a hybrid between Sporadica and Lemannii, and possibly at the second population with Redacta. And so we did a microsatellite analysis and found that, yes, indeed, these are the two parents, and it's a classic F1 hybrid that's just been perpetuated by having a clonal reproductive system. So it's now been removed from the threatened species list, but it's actually in national parks, so it's actually quite protected anyway. Just put this in to show, because Rob mentioned it yesterday, Eucalyptus phallacis, it's the Mealup mallee. Was suspected to be a hybrid more recently, but we weren't. Decipiens was suspected to be one parent, didn't know what the other parent was. And then more recently, somebody found a little patch of Eucalyptus virginiae just down the slope towards the beach from this patch. And so we just recently done a genomics study, and you can see here that you've got Decipiens and Virginiae, and phallacis is a classic F1 hybrid with half the alleles from each, and on a PCA it just sits right in the middle here. So again, that's just found what the other parent is for that. So we heard about species phylogenetics and species relationships at high levels yesterday. I've done more work at the low level because that can be particularly challenging, just understanding species boundaries in eucalypts. So here was an early study of Eucalyptus angustissima. This group of species were used as being looked at for oil mallees, in the oil mallee industry was getting underway in WA. And so here, subspecies Corenda here of Visma was thought that might actually be a separate species, and we did a population-based kind of phylogeny and found that it wasn't, that it was polyphyletic, and indeed we've now raised Corenda to be a separate species. More recently, got evidence of cryptic speciation in Eucalyptus salubris. So Eucalyptus salubris, that's gimlet, is a lovely coppery bark. It's widely distributed through the semi-arid zone in WA, and we were doing an adaptive genetic study on this species and came up with Dot and Brad and Renee, and came up with two lineages. So here's lineage 1 and 2. We originally only had 9 populations. We've done another, brought it up to 22 populations, and also included 2 populations of Eucalyptus ravido, which is a related species. The only difference is it's got glaucous stems, so it's a glaucous form, but recognised as a different species. And it did, so we kept the same 2 lineages. They're also differentiated by specific leaf area as well, in the opposite direction as to what you would expect from responding to climate, which is interesting. But you can see there's no geographic separation there. They're quite overlapping. And Eucalyptus ravida clustered here, they're these diamonds here with lineage 2. So the difference between the 2 lineages is greater than the difference between the currently recognised species. So we're actually now currently looking at doing a broader phylogenetic study with other species in the group to get an understanding of the broader relationships amongst this group. And Eucalyptus virginiae, just coming back to that, why we started actually looking at that is that this patch that was found just down slope from the Mealup mallee is a mallee patch, and it turns out there's 3 patches in this, so it's 3 individuals. But the Eucalyptus virginiae, what was previously recognised as Virginia, which was split out of Lanepoolii just a few years ago, was actually a tall 40-metre tree growing near Denmark. So here's the little map. So we've got the Mealup mallee up here, and we've got the rest of Virginia down here. So that's a 250 kilometre difference. And so how similar are they? And again, we've got a mallee and a tree form. So when we look, we also added in Decipiens to get the relationship for phallacis to Virginiae, and Lanepoolii and Relicta, because Virginiae and Relicta were pulled out of Lanepoolii at the same time. When you do a PCA together, they cluster into the species quite nicely, but when you do a PCA just in Virginiae, you get the differentiation as you might expect from the disjunction with the Mealup population here, But then this little population's thrown a spanner in the works because it's 5 trees that's only about 30 kilometres away from the main populations in Denmark and Mount Lindsay, and yet it's differentiated as well. So something a bit odd going on there, which we've got to do a little bit more work about. So that brings me to the tree kind of versus mallees, and how good is that as a taxonomic character, because there's a bit of debate about that amongst taxonomists. This was an early study of Eucalyptus loxifleva, which has 4 subspecies. 2 are trees and 2 are mallees. And so in a maximum likelihood phylogeny of RFLP data, we've got the mallees clustering together, got the trees clustering together, except for these 2 populations that come out early on. But when within the mallees and within the trees, you don't have separation between the 2 subspecies. So it does look like there is some understanding that mallees are related, the trees are related, but not within that. More recently, using genomics, we're looking at the difference between lignituberous versus non-lignituberous species, so that is that a valid taxonomic character? So this is a maximum likelihood tree, and this is an ancestral tape tree. So we were looking at 2 pairs of species. So Eucalyptus gardneri and Eucalyptus pluricollis are morphologically identical, except one has a non-lignituberous as a tree, and the other is a lignituberous and a mallee. And also denser, the 2 subspecies of Eucalyptus denser, the same situation. So we looked at those pairs with the other species that are in the subseries Levispermi to see how they were related to each other. And we would actually expected that if it was just a gain or loss of a capacity to form a lignituber, then we would expect them to be sister taxa together. And what we found is that they were not sister taxa. I appreciate you probably can't read this. They're all colour coded, so you can look at the colours, and then the names I've just put them down the set. But the 3 tree species are up the top here, and they cluster together in the dark blue, the light blue, and the red, the 3 species. And then we've got this clade here, which is all the mallee species. Sometimes they cluster together, like Redunca here, and other times they're quite dispersed throughout the tree. So it looks like there's something still going on amongst the mallees that we haven't got to the bottom of the relationships between those, but the trees are coming out as clear, good taxa and separate. So lignituber does appear to be important taxonomic character, and we're just going to do this in another group as well to do it. If you actually plot the ancestral lignituber as an ancestral character on the tree, you actually get the non-lignituber state obviously as the ancestral state within the subseries, but once you go to the higher levels, then you don't get it. It becomes equivocal, so it looks like it's probably multiple gains and losses throughout the relationships in trees within series. Okay, another area that we've done quite a bit of work on is phylogeography, and so using chloroplast markers, so recognising that chloroplast markers you can share between species, but if you're looking within a species, it's generally pretty good to understand the evolutionary history, and when we've compared the evolutionary history of eucalypts to other species in the same landscape, we're getting the same patterns, so I think it's giving us a good understanding of looking back into evolutionary history. So here's an example again from Mary throughout the southwest, and look, we didn't expect this level of diversity with the haplotypes. You've got a cluster of related haplotypes here, but the divergence amongst these haplotypes is one of the largest I've ever seen in all the studies that we've done on various species. And if you look at it, so these divergent haplotypes here are all clustered in the centre of the distribution. These ones are off to the north, and then these more related ones down to the south. So we think it's probably a centre of origin here, and a branch gone up to the north, and then it's expanded out into the south. We get a similar pattern of expansion into the south in 4 other species that we've looked at in the forest tree, Bossia canedia and Alocasuarina. So it looks like these forest areas opened up probably about 300,000 or 400,000 years ago, and these species have been able to move in and colonise them. If you're looking at phylogeography, often you're looking at, so we get a lot of signals of persistence in the landscape, persistence through long time frames, but you can also look to see, well, are you getting patterns of refugia? So it's often what a lot of phylogeography looks for. This is Eucalyptus leucoflora in the Pilbara this time, and it occurs through the Hamersley and Chichester ranges, which are through here, and then occurs also out onto the flat areas around it. And in looking at the haplotype and network based on chloroplast sequences, you'll see most of the diversity of the haplotypes are present in the Hammersley and Chichester ranges, which is consistent with them, with ranges being a refugia in arid environments, and then being able to move out onto the lowlands at later times. Interestingly, in Eucalyptus wandoo, we weren't expecting to see a pattern of refugia in wandoo, but we found a lot of diversity in this central region here. So here's the network. This is where the distribution of the haplotypes is. No obvious reason as to why you'd have a refugium in that area, but there's quite a lot of diversity here compared to lower diversity to the north, east, and south. When we've done an SDM, so spatial distribution modelling at the present and at the LGM, you find that the diversity is all in the area that has remained most suitable at the LGM. So it looks like that's influenced the persistence of the species in that middle area, and then there's been more expansion out. It's actually consistent with the drying, the decline of Eucalyptus wandoo on the eastern margin. That's probably expanded out more recently and is now contracting back again with changing climate. So that's just kind of a summary of all the various ways that we've looked at different kind of combinations and understanding of genetic diversity with molecular markers in eucalypts, and getting to understand the really high levels of genetic diversity they've got, low population differentiation, mixed mating systems with high gene flow primarily through pollen dispersal, clonality through lignotubers, being able to understand close species relationships, because we can't do that in a more rigorous phylogenetic framework, and understanding phylogenetic patterns, which are really indicating the persistence of species through the Pleistocene. Most of the species that we've looked at have all shown persistence right back through to 2 million years ago. So then that leads me to looking at, well, what are we going to do in the future? And that's what we've got more into, looking at, well, can we, so particularly with the power of genomic markers, looking at adaptive variation. So the first study we did was teamed up with Dot and Brad and Rene, and we looked at 3 widespread eucalypts, Eucalyptus tricarpa in the east, and then two species across the semi-arid region in the west, and looked at, did DART-seq analysis and outlier analysis, because that was what we did 6 years ago. That was the way to do it, and also looked at morphological traits and ecophysiological traits as well, to get an understanding of how that relates to the actual traits that matter on the ground. So this is a very busy slide, but essentially what we found outlier markers. So essentially what you do in an outlier analysis, you're just looking at the baseline level of FST, differentiation across the climate gradient, and then looking for ones that are significantly more differentiated than that. So we found outlier markers in all of the species, and we found outlier markers correlated with climate in all of the species. And some of the correlations were quite strong. So here's some examples of the correlation of allele frequency and climate variable, much stronger than we expected. And even in Eucalyptus lubris, where we found the 2 lineages, so we were only able to do it on lineage 1, which meant we had a much shorter climatic gradient to work with, we still found signals of adaptation across that gradient. Because, of course, all of the DART-seq markers were mapped to the Eucalypt genome, we were able to plot them across the genome and show that it was pretty representative across the genome. All of these little dots here are the SNP markers across the genome in each of the 3 species. And then these are the blobs, the circles are the outlier loci. And you can see there's a bit of a cluster up there, but otherwise they're distributed across the genome. And we did find 3 cases where there was multiple evidence of SNPs linking to the same genes in more than one species. I won't go through a lot of this, but this is to show that we did have the morphological and physiological traits, and morphological traits were showing to be adaptive. We had a common garden experiment in both ends of the gradient for tricarpa, and only at one end of the gradient for Loxifleba, and in those common garden experiments we found the evidence for adaptive morphological traits and plasticity in physiological traits. We didn't have that in Eucalyptus salubris, but what we did have, the year that we collected it, the rainfall trend was the complete opposite from the long-term average. So the southwest was in drought, so it was dry when it should have been wet, and there were several rain-bearing ex-tropical cyclones come down in the east, in the northeast, which made it wet when it should have been dry. And so so this is a delta 13 carbon, or water use efficiency, is clearly correlated with strongly correlated with the last 12 months rainfall, and showed no correlation with the last 5 years. So that's certainly in physiological traits. They can respond very quickly when there's water available to them. More recently in Crimbia calophylla, this is work with Colin. He's going to talk about this later today. So done the same kind of thing, but using an environmental association analysis this time, and found 609 SNPs significantly associated with environmental variables. So we've just got 3 plotted here, one for temperature, one for rainfall, and one for aridity index. And on the left-hand side is the climate variable in each case, and on the right-hand side is the allelic turnover of the variable of the SNP that's related to that climate variable. And you can see even over relatively short geographic distances, you can get strong turnover in allelic frequency. And here's the spline out of it. So we've got strong signals that we're getting adaptation to climate across these distribution. Have looked at similarly in restricted and widespread species. So if you've got less climate variation, are you going to see less signal of adaptation? And we've done 2 sets of pairs of species here. So a widespread species and then a phylogenetically related species nested within that distribution, 2 in South Australia and 2 in Western Australia. So here's the plot of on the left is the South Australian species, and you see they've got similar levels of numbers of loci associated with climate. And this is the sympatric zone here, and the sympatric zone has much less compared to the restricted species. So that's not what we were expecting. Whereas in Western Australia, we've got the widespread species and the restricted species, the widespread species with more loci than the restricted one. And if you just restrict the widespread one to the sympatric region, you've got similar levels. So you're getting a signal there of, as you'd expect, if you've got a greater gradient, you get more adaptation across that gradient, but something different is going on in South Australia there. You would have seen Zhao's study, I hope, last night in the posters. So he's done the similar work to calophylla, and we're going to do that more comparison. But here's another bit of work also in Jarrah, done with Justin Borovich and James Kondiolis, and just showing that you can use so we took transects across the distribution, 4 or 5 transects across the distribution, most going east-west, one going north-south, looked at the genomic variation and then incorporated into general dissimilarity modelling of the change in climate and the change in allele frequency that you would need to match the change in climate going out to 2050 for a moderate climate scenario. And you can see that you need greater change in the wetter part of the distribution than in the northern part of the distribution. And that's giving us help to understand how are we going to actually respond, and what kind of perhaps assisted gene migration or whatever do we need to put in place to make the forest resilient for the future. So just to sum up, that's a summary of a whole range of ways that we can use genetic markers from back to where we started with allozymes sort of 20 years ago, through to genomic markers that we're using now, range of understanding the diversity and patterns within eucalypts, and then looking at signals of adaptation. So how are we going to move forward? How are we going to help these species persist in the landscape with changing climate, and are there things that we can do? Or are there species that we don't actually need to worry about because they've got the standing adaptive variation and they're going to be able to manage into the future? And I'd just like to acknowledge a whole range of people who've worked with me on a whole range of these projects, both on the genetic diversity studies and in the adaptive variation studies, and of course recognising some of the funding that's been provided for these studies. Thank you.
Okay, we have time for 2 questions. Yep.
I was interested to see the clonality and lignotuber as potentially fostering some traits at some level. Do you think there's climatic drivers that are determining those shifts in the traits as well?
We have plotted pairs of lignotuberous and non-lignotuberous species that are closely related, only differing by a lignotuber or non-lignotuber across a range of not just these species but a range of ones, and the In all cases, the non-lignitures, the tree species, were shifted more to the arid end of a gradient than their mallee presumed close relative. So it does look like there is a climate effect there. Yeah.
One more quick question. Jack?
You mentioned the F1 hybrid being delisted, and in that case it was conserved anyway, but I was just interested in your ideas on whether we should be conserving hybrids.
Yeah. So in WA, under the legislation, you can maintain a species on the threatened species list if it meets 3 additional criteria. It has to be naturally occurring, it has to produce seed, so it has to be reproductive, and that seed has to be morphologically contained within the current species, the taxonomic definition of the species. So that's essentially saying if it's a long-term hybrid hybrid speciation, so it's speciated into a new species, then we'll recognise it. If it's an F1 hybrid like this, then we won't. And in this case, it doesn't segregate. No, actually, we haven't got seed from this species. It doesn't produce much seed, and it doesn't, so we can't check the segregation. But from the genetics, it appears a classic F1 hybrid, and so we wouldn't keep it on the list. So we do recognise if it's a hybrid speciation event, but not if it's just an F1 hybrid. In this case, just being perpetuated for thousands of years by becoming clonal.
Well, thanks, Margaret.
Thank you.
Okay, our next speaker doesn't really need introduction. It's Brad.
Well, I'm not sure if I'm going to disappoint you, but I'm not going to mention molecular markers at all. I'm going to stick to a basic old-fashioned ecological genetics, quantitative genetics approach to our understanding of population differentiation and evolutionary processes that we see in our eucalypts. And in Tasmania, we've been working on these issues for many, many decades, and I just thought I'd just flip through the decades and give you a glimpse of some of the amazing systems that we have outside these walls and some of the issues that we've addressed through time, and just trying to sort of relate them to some of the issues that we're facing these days in terms of actually dealing with climate change adaptation. And so going back right at the very beginning, back in the 1950s, and really a lot of the work done there was research. Professors Barbara and Jackson here and Lindsay Pryor in Canberra, and really they were really focusing on the adaptation of our eucalypt populations to the to the altitudinal gradients. These were the obvious things we have here in our mountainous Tasmanian terrain, and really every time you got to a mountain and a species transgressed large areas of altitude on those mountains, you would find phenotypic differentiation in the wild, and progeny testing would show that one way or another you would get genetic differences associated with those clients. And this is the time when we actually started developing the paradigm of eucalypts exhibiting a really close adaptive response to the environment. And really my pick of the studies of that time is this one here of Eucalyptus sinigera, one of our endemic species, which as you go from just at the mountain out the back here, as you go from about 600 metres altitude in the really closed wet forest at the base of the mountain to the up to about 1,000 metres in the subalpine, open sort of subalpine forest, you get a really dramatic change in phenotype. In the closed forest, you've got all the seedlings and the adult trees, the leaves are shiny green, and very rapidly over a 200 metre transition in altitude, you get a step climb to foliage which is highly glaucous, covered with a waxy layer of glaucousness, and it's occurring, it's got a genetic basis to this climb, and there's a whole lot of other traits which change over that very short distance. But what it really shows is the power of natural selection in really steep environmental gradients to shape the genetic diversity that we see in species. And this is just really lovely work, and we're still working on these things today. But if we slip through to the 80s, and this is when I came on the scene, and we actually started moving a little bit more into the idea of things changing, and in terms of at least space. And there was a couple of points that emerged from those studies. Firstly, the studies by Neil Davidson is really emphasised the importance of rare climatic events, extreme climatic events in shaping the distribution of our species boundaries and whatever. And he, for example, looked at the consequences of a -22 frost in the subalpine plateau off the edge of Mount Wellington. That was the lowest temperature that had ever been recorded in Australia at that point in time. And just the effect that that had on the species boundaries was really quite dramatic. And the other thing is at this time, followed by the frost, then it was droughts. And so we hit a whole series of droughts at this time. We thought that they were bad, but we had actually seen nothing compared to what we see these days. But I was doing my PhD, looking at the regeneration and dynamics of a hybrid zone between two of our really closely related eucalypt species, Eucalyptus risdonii and amygdalina. Really closely related but very morphologically different, and the hybrids are really easy to see. It makes life really easy when you've got really good morphological markers like this. And everything that I looked at in that hybrid zone, the reproductive output, the growth of the seedlings, the established plants, everything was telling me that the risdonii was expanding its range into the amygdalina. And some of the things that came out of that was that firstly that the seed dispersal was a really limiting factor to the adaptive response of our populations to any environmental change. Like the seed here was just falling virtually straight to the ground. There was a bit of asymmetry, downwind flow of seed, but apart from that it was very restricted. But the one thing that we did see was that there was hybrids right out from the boundary. They were in the open pollinated seed, and they were established as saplings and seedlings and even adult trees well out from the boundary. So we imagined there was a wave of hybridisation spreading out from the expanding population, and this is when we developed the idea of hybridisation as a dispersal mechanism, and that peat species could be resurrected from segregating hybrid swarms, which received a bit of flack at the time, but I'm sure history has proven us right. But anyway, so the 1990s we started, that was significant in two senses for us. That firstly, I reckon it was our first signal that climate change was starting to impact our gene pools, and we certainly saw one of our subalpine species, Eucalyptus gunii. It's one of our most frost-resistant eucalypt species, and the most frost-resistant component of that species just died. And so it started early 90s, and by the end of the decade the main core of that, it's a subspecies now, was dead. And this really started to get our focus onto climate adaptation in our eucalypts. And the other thing that occurred at that stage is that we started shifting into studies of quantitative genetics using very large-scale field trials. And I just want to run you through a couple of our key examples of the study systems we've worked with. Eucalyptus globulus, that's the commercial species. It's the major temperate eucalypt planted in regions around the world. We've got an endemic complex here, again involving risdonii, but it's a closely related species, Eucalyptus tenuramus, and Porciflora, which is one of our eucalypt species which has the biggest altitudinal range, certainly both in Tasmania and on the mainland, and it's now a major restoration species in our dry midlands. So if we just look at our work on Eucalyptus globulus, I reckon that there's not many species of eucalypt which we know as much about the provenance variation in quantitative traits as globulus. We've been working on it for about 30 years and we're still going. But really our knowledge of the phenotypic variation, the functional trait variation in this species comes to this amazing collection that was funded by the forestry companies nationally and internationally to do for the CSIRO Tree Seed Centre, to do a really big collection of provenances and family seed lots of that species. So that was the really the basis of the insights that we gained on this genetic structure of that gene pool as it went from the mainland, coastal regions of mainland, across the island populations in Bass Strait, big continuous populations down the east coast of Tasmania, little disjunct ones on the west coast. And so the seed from these collections were distributed all around the world and across Australia, 76 international trials, 26 Australian trials, and our focus was on the Tasmanian ones. We didn't have far to go. And but they were really the best ones. So they were, had all the seed lots, about 600 seed lots in them. The whole collection was in Tasmania, and so we did a lot of work on these trials. But also we obtained data thanks to Greg Dukowski, who did some work in Western Australia at the time, and really some of our best information on the climate adaptation in globulus comes from the Western Australian trials. And really it was an unfortunate drought that had some quite severe impacts on the plantation estate at that time, back in '93. But this severe drought hit all of these field trials in Western Australia, and Greg scored, was it Greg, 22,000 trees for their drought damage at the time. And this allowed us to get really good insights into the spatial variation in drought susceptibility of Eucalyptus globulus across its range. And you can see here on the west coast, all these west coast populations really drought sensitive. The bigger the circles, the more drought sensitive. The bigger the triangles, the more drought resistant. So we had very good, strong differentiation in drought tolerance in our species. And of all that, we had sort of gradients going in the continuous populations on the east coast of Tasmania, from the wet areas down the south here to the north. Within the Otway Ranges, for example, we had going from west to east, another gradient in drought susceptibility, running down from Gippsland, from the high altitudes down to the coast, another gradient. And the thing that, there's a lot of climate correlations in all of this, but the climate correlate that really did sort of match best with all of this, both the global variation in drought damage and the local patterns of variation, was the temperature seasonality. So whatever it is, we suspect this is pretty good evidence that it's climate-driven adaptation. And the beauty of these field trials is that we actually have pedigree links. Because of the same families are grown in Tasmania, they're grown in Western Australia and Ethiopia, we can do genetic correlations between drought damage in Western Australia and the traits that we've measured in Tasmania in the field trials. And Greg did many of these correlations, testing all our traits against what was correlated with this drought damage. And really the things that came out were wood density, and we've got plenty of evidence to suggest that that's a good mechanistic basis to that with the xylem embolism. But the thing that really did surprise us a bit was an even stronger, slightly stronger correlation with bark thickness. Now, exactly what this is about, we're not sure. It could be due to the indirect relationships with wood density, or it could be due to the co-occurrence of drought and fire. Bark's normally associated with thick bark fire resistance. But I do suspect that it's giving us a signal that the bark is actually a component of the mechanism associated with drought susceptibility in terms of water storage and non-structural carbohydrate storage. There's a bit of work coming out on that. The second study I want to talk to you about, and I've chosen this because it's quite interesting in terms of the evolutionary process which it demonstrates. And really, this is the work of Bob Wiltshire. He started it during his PhD in the early 90s, and he was our photographer yesterday. But really what we're dealing with here is the process, a very important evolutionary process, I think, which is the capacity of our populations to change the timing of developmental events. It's called heterochrony, so it's heterochronous evolution, and it's a way that populations can make rapid changes to their phenotype with minimal genetic restructuring. It's just the timing of the onset of certain characteristics. And we've got a beautiful example on the eastern shore of the Derwent River here, where we've got Eucalyptus risdonii here, which is a homoblastic species which effectively retains the juvenile foliage into the adult stage. It's called neoteny. So it's a bit like the salamander and the frog story. It's occurring all through the animal and plant kingdom, these processes. But the related species, very closely related species again, very similar seedling morphology, but it is heteroblastic and it develops the adult foliage early on. So this occurs, we've got a very steep gradient in that time of that onset of the heteroblasty just in this very small area on the eastern shore. And it's got a genetic basis. This is the results from field trials showing that it's under genetic control. And when we look at the climate space with which this homoblastic juvenile form exists, what we find when we model it with random forest here is that sitting up there in a little corner of the climate space. It's that area which is really extreme in terms of arid and high temperatures, and so it's a very extreme component of that environmental space. Now, we don't know, we've got no information on the intensity of selection against the early phase change form in the dry environment, but we do have some idea of the selection against the juvenile form in the wet forest situation where we actually had our original field trial. And so we can actually do a selection analysis and to see what the selection coefficients are against this trait. And at a population level in these trials, we can actually see that certainly at the population level, the juvenile forms are selected against. But what's really exciting is when we get down to the variable intermediate populations, we can actually show that natural selection is acting against the juvenile forms within a population. So it's evidence of selective filtering of these intermediate populations. And so it's the first signal that, yep, we've got a direct fitness value to this trait. Now, we can go on and then model the predicted selective surface acting on that trait under climate change. This is the end of the century. This is, for example, our distribution of the trait in the current environment. If we assume that the adaptive surface and the selective surface are in equilibrium in the current situation, then this will change into this selective surface in the by the end of the century. And what we see, first of all, is what Margaret was mentioning, that the impact of climate change on this trait is not uniform. There will be parts of the distribution where juvenility will be favoured. That's the yellowing here of this part of the range. But the area where the homoblastic form, under this climate model at least, is suggesting that the delayed juvenile foliage will be selected against. And so, you know, there is issues there in terms of actually trying to actually translate these changes that we predict through climate models into actually realised impacts on the fitness of the traits. And this is where field trials come into play. We can use space for time to try and understand the impacts of these changes in the climate. And so you can see, for example, that the difference in the future between the red here and this yellow here is small compared to the translocation that we've already got an indication of the selection coefficient on. And so this just moves us into the last phase of our work in terms of our latest range of provenance trials. It's working on Eucalyptus pauciflora, where we've actually done a very large 37 populations from throughout the Tasmania distribution. 5 to 10 families are being collected per population, and we're just rolling these out every time we get a chance to put a progeny trial in right across the landscape, with the idea that we can understand the adaptive differences between the populations and also get an idea of the how we source our provenances for the restoration work that's going on. And just to give you it's early days, but just to give you some idea of the sort of thing that we're looking at, we're trying to actually model the transfer functions across environmental gradients as best we can. And it's certainly, the signals are fairly weak at the moment, but what we are testing, this for example shows what happens if we take a provenance from a home site estimate of 24 degrees C and we move it down to a cooler site here, and this is the growing period, that's the trial site prediction of the climate there. You can actually look at the relative response. We move colder ones to that warmer sites, etc. And we can see in this situation that the warmer populations, provenances from originating from warmer areas are actually growing better than the local at that situation. If we go up higher though, these low altitude warmer derived populations are then starting to drop off in their performance. And so this is, in the future, will give us an idea of the amplitude of the transfers that we can do and we can expect from climate change. And so really the future challenges we have is really to get an understanding of these provenance transfer functions, to guide provenancing strategies, but also I think you can look at it in the reverse case, because they will give us an indication of the adaptive limits of which our native populations could withstand changing temperatures. The other issue is the functional traits associated with this variation in fitness, and is there an adaptive syndrome that we can use for the future? And I suspect it might be dense wood, thick bark, and probably lignotubers. And then if we look at the adaptive capacity, and this is really where I see molecular markers and genomics coming into play in the future, is really trying to monitor the change that's occurring, the evolutionary change which is occurring in our local populations at the population level. It's really intractable with quantitative genetics, with long-lived forest trees, to get at this information of genetic change in populations, and seeing the impact, being able to monitor the impact of climate change on our populations. We really do need good, validated, adaptive molecular markers. Okay, thank you.
Okay, we're running a little bit beyond. We probably have time for one quick question. Yeah, thanks.
I was just really curious with the Ristonii tenueramus sort of complex there. Thinking from a conservation point of view, if those sort of Ristonii are going to get pushed out and be selected against in the future, what does that mean, and is that important if that genetic variation sort of exists in tenueramus anyway? Like, do we need to worry about conserving Ristonii, or this is just one of those changes that will occur in the future of them fading out in tenueramus?
Certainly. There wouldn't be the full range of genetic variation within the tenueramus populations to achieve that. There is in the intermediate populations, which is where we had the selection going on, but certainly not in the main core of the... it'll take several generations to actually evolve a Ristonii out of the tenueramus up north. Yeah. So really, and the other thing is, like, are those climate change differences significant enough to sort of that Ristonii won't have time, either plasticity or time to adapt to those changes?
Maybe time for another question.
That's too quick.
Yeah.
You mentioned plasticity.
Yes,
so
yeah.
In those traits that you've obviously got multiple trials, say for example the wood density or one, do you see lots of plasticity in that trait, or is it a fixed structural trait?
We certainly, there is plasticity in the trial means, but the ranking of the genotypes in those trials for wood properties is incredibly stable. Growth's another story altogether, but you certainly get wood density increasing on dry sites versus wet sites, for example. But the genotype GYE is pretty good.
Okay, thanks, Brad. Next is Justin Borovitz. Justin did his PhD in California, went and worked for a while in Chicago, the University of Chicago, and has been here since 2012 at ANU.
Thank you. It's a pleasure to be here. I want to try to look forward 20 years, sort of give the vision, the high-level vision that Xander and Jerry Tuscan started yesterday. That's a wonderful historical perspective that Brad set out. We know that climate change is coming strong. It's going to hit agriculture and ecosystems, and the proposed solutions like bioenergy with carbon capture and storage, and also this large-scale afforestation, are both displacing agriculture and ecosystems. And so, what can we do about it? Eucalypts can help. That's the short answer. So let me just say that we're hitting this really triple threat. Even providing enough food is difficult, and conserving endangered species is getting harder. And just shifting to renewable energy is no longer sufficient, that we need to pull carbon out of the atmosphere at high levels. And we're seeing unprecedented droughts, land clearing, and this blue line here, it's hard to really take home what we mean by carbon drawdown. Now there's an X Prize coming out for this, and different challenges. It's essentially 1 trillion trees. It's a column of wood to the moon that's the size of this room. It's hundreds of cubic kilometers of fertilizer, if you will, for trees. So we need to move quickly. the land sink is the largest uncertainty in the land system, and that's an opportunity if we can get this right, that basically the drawdown that nature is responding to the changes is compensating for our land use change, but it's in no way makes up for this fossil. So basically we have to shift our energy systems, but we also have to really engage the whole land system to be able to do this. And what we're seeing already is these intensifying droughts, fires and flood cycles. And I was just flipping through the Nature Handbook here in Tasmania, where just 10 years ago it says most of the state is too wet to burn, but we're into new cycles. You would have also said that for Canada and Alaska don't generally burn. We're in a new state. This Anthropocene is going to be heavily managed and heavily modified. So how do we get ahead of this is what I want to talk about. And the answer is not what we've been doing, which is more land clearing, rivaling Brazil, but it's regeneration of these degraded lands. So essentially we're already heavily modifying with agriculture about half of the planet. You can think about 10 billion habitable hectares. Half of that is in agriculture. Now the other half is sort of natural forests or managed forests, and we really need to think about this intersection about how we integrate rather than the standard contrast between agriculture. So really there's 1 to 2 billion hectares of neglected or marginal land that has been the target for this kind of bioenergy, and it's not that productive, so it's hard to get a lot of bioenergy out of these areas. But if we can shift our practices, and eucalypts are fantastic at growing in these harsh, rough conditions and resisting fire and improving the landscapes, then we can start to get to that gigaton sort of scale. And this is $1 trillion a year global opportunity to address this if we do it right. So how do we make more land in the world? We have to go vertical. We have to think about multiple layers for food, carbon, and biodiversity habitat. And we can think about layers below ground for deep-rooted perennial grasses and trees, and layers above ground where you could have grasses and trees, and integrate these three spheres that are often discussed separately. So I'll go into some modern evolutionary genomics that fits into this meeting. I'll talk about some of the phenomic technologies that we're applying, drones and arboretums, and then try to talk about the regenerative agroecosystems from the farm to the forest to the planet, where it will actually make a difference in the climate. So when we set out these trials that Brad has been a real pioneer on, we need to think about how to lay them out in the earthworks and the water so that they actually can get established, and some renewable energy to pump hydro irrigation, and a whole data system that every kind of farm, modern digital agriculture farm, needs this infrastructure, a build piece, and then a management piece. What are the plants, grasses, and trees? What are the microbes? Obviously for acacias we've got nitrogen fixing capacity and mycorrhizal fungi. These are important for the capture, the growth of the plants and the biomass. And then there's storage. How do we make that permanent? How do we use biochar as kind of a charcoal to improve the soils and fix the biomass for the long term? And then there's below ground, there's rock weathering, which is also a long-term carbon store. Now, we need to put this together in big models, and we've been working on the genotype-phenotype problem for a decade now. Jerry talked about that yesterday. We can reverse that with genomic selection, so we can sequence a line. It may never have been in a trial, but it could be related to something that's been in a trial. Therefore, we can know what the genetic variation is. We can know what it'll look like, how fast it'll grow, what its climate envelope would be. We'd like to do that with G by E. We can also do what Margaret showed earlier, assisted migration. We can move things into their future environments, and we can look at the genetic relatedness of things today and things in those future environments and make a genetically informed migration decision. This is not adaptive per se. This is just sort of assuming that local adaptation has been the rule, and we can try to move things into their future and climate. And crop modeling and plant growth, we can look at how the environment, and especially as the environment gets more unpredictable, the genetics piece has less influence. But we have great models now to take the weather station data and predict growth and crop yield. And there's been decades of training and tree models to be able to make these environmental predictions. So what's the third generation? We showed this yesterday. This is whole genomes. This is going to get away from the biases of looking at a few genes in the chloroplast or a few tens of thousands of SNPs and where everyone's study is uncomparable. Once we have whole genome data, this can be done in the pocket or in the field, we'll be able to make these comparisons. You might say, how many species are there? Well, how many species of dogs are there? And I like to think about breeds or races because they really are intermixing. And the diversity that we're starting to see, these are a list of a dozen or so new reference genomes that we've just put up into NCBI. See, Scott Ferguson's got a poster on a new Meliodora assembly. We had 100 megabases overnight, so this is coming along. This is the quick preps. It's really exciting new technology. Here's Scott's assembly. So the contigs, 400 scaffolds came out. The grandis is actually beautiful, 11 chromosomes, but there's a whole bunch of things that didn't make it into the assembly. So we've got a similar sized genome, 640 megs. That's the real challenge now with assembly is to actually assemble the independent mother and father. So Rob yesterday was saying it's really a gig-sized diploid genome where the two parents are 4% different from each other. Like, that's just astounding. That makes maize seem like an inbred, right? And maize is the diverse crop that we have. It's widely adapted to the... So in mammal terms, 4% diversity would be like if your mom was a cow and your dad was a sheep, all right? And that's compatible. And then the offspring are just whole new species wandering around and filtering into different environments along altitudinal gradients. So this is what we see with sequencing in genomics now. And the phenomics, this little video is not playing, but this is a 3D point cloud of the tree. This is the CSIRO, and Linda will talk about this, is across the ANU gate. There's a sort of a legal barrier, but the scientists like to take seeds across and collaborate, and so we chose this tree as the reference Meliodora tree with that symbolic reason. And we're growing these in climate chambers, climate capsules I call them. They're energy efficient little boxes where we can simulate local and future conditions. 50 shades of green with hyperspectral imaging and lots of impressive new tools for phenotyping seedlings. In the field, this is the National Arboretum, and my postdoc now, he's the director of the ANU node of the Plant Phenomics Facility, put in what he calls the Phenomic and Environmental Sensor Array. This is just camera towers and weather stations, and it's actually quite cheap to put these in. It gives us leaf-level resolution, giga panoramic images of the whole arboretum, and now drone flights monthly can be stitched together into three-dimensional point clouds so we can see the size, shape, and color of every tree as they grow and develop. And then that gets put into computer models so you can be in virtual reality and run around into Ethiopia. So even though it's not out here in your backyard, you can go remotely into these trials or into predicted future trials that we'd like to put in and see how the trees would grow. We can look at soil moisture, dynamic effects of how the plants would respond to both agronomic treatments and extreme weather that's coming. So this is sci-fi virtual reality, and I think it's better than not having the data. It's the way to communicate these to large-scale restoration planners. And how do we get it done? There's a new company. There's quite a few new companies. One of them is Biocarbon Engineering that does drone seeding. So once you have the right genotype, the right maternal line, which could release huge amounts of cryptic diversity, especially these hybrids, we put it into the landscape, the right maternal line and the right contour row, and in the right environment for the future restoration. So matching the plants with the microbes for carbon capture, and then making that permanent with management of nutrients and charring. So these digital farms is a proposal that we put together as a team at ANU to try to solve some of these grand challenges. We want to maximize light, input, and actually the dense forests are light limited in many cases, especially in Tasmania where you get more water than we do in Canberra. We want to really maximize water use efficiency, minimize evaporation, make sure that the rain gets into the soil. We have a lot of compacted, degraded land where the water just rushes off. You see these Grand Canyon-sized erosion gullies that can be seen from satellite. Like, there's a lot of opportunity for improving the land so that the trees can grow. And then ultimately, we've got these biological machines programmed by their genetic code to capture carbon. It's got to compete with the Elon Musks of the world that have these carbon-sucking machines. This is a biological machine that regenerates itself. It's amazing. How do we put these pieces together in this farm? This is a proposal that's almost a year old. The politics of land use is quite challenging compared to all the other technologies that we work with. So really, I think engaging with progressive landowners, and Margaret McKinnon's here working with us in the Upper Snowy Land Care to try to look at landscapes that could be for agroforestry or for more ecosystem regeneration. There's certain areas along the creeks where there's remnant trees. That's where we should be preserving the biodiversity. The riparian areas, the alpine areas, and then slot in the agriculture into the steppes in between. So this was a concept that's been actually deployed in China at a huge scale. The lowest plateau, if you're familiar, that's a huge area, like 1/3 the size of Mongolia up on the river. It makes the Yellow River. It used to run yellow with all the lowest sediment going into it, and they've transformed that with these agro ecosystem steppes, and they've also recolonized these sand islands. So there's a movement underway, and my postdoc Helen just flew back from the U.S. and she showed me, even if you look down, there's terrace forming going in the U.S. So this is happening, and we'd like to do that on the scale of the Monero, for example. This is 1 million hectares. These are large enough kind of agro-ecosystems that they're beginning to draw down sufficient carbon. Now, we've mapped the landscape genetics. So this is work that I did with Linda and Margaret and others, led by Megan Supple, a former postdoc, where we looked at Meliodora across the range. And in this case, we found a few kind of cryptic hybrids that always pop out, mistyped lines, and then a huge amount of wide crossing. Like, I've never seen such a well-shuffled population structure. Here was less than 1%. We could hardly explain anything with the first two principal components and individual diversity. Also, we did see some of these yellow replicates. This is what Margaret also mentioned. These clonal trees, sometimes 10 meters apart, are actually the exact same tree. So as we're collecting maternal lines, sometimes they're not individuals. But if you map that onto the landscape and then try to look at not individual adaptive SNPs, we really need more and whole genome sequencing for that. But if you look at background genetic diversity and how that relates to geography and environmental isolation by distance and environment, then we can make projections about how the current geography and environment relates to genetics, and then forecast that to look at what are the zones. And maybe it's not surprising, the big dominant drivers are rainfall and temperature. And so the tropics are kind of moving down. The summer, winter rainfall zones are changing, and that's kind of the major signal that we need to think about as we're shifting provinces. The internal dry areas are continuing to be internal and dry. So let me kind of breeze through now at the highest scale. The simple concept is plants are drawing in carbon dioxide, okay? They need better water and nutrient use efficiency to maximize that in landscapes that are regenerated. To not lose the carbon, this is the biological carbon cycle on land actually shifts 120 gigatons of pure carbon through the vegetative system. Most of it comes right back out in the cycle, which actually dwarfs our anthropogenic cycle. So there's a big opportunity. The top generals in Canberra saw this slide as the most exciting for environmental security. They realized that our human system is not actually dwarfing the planet. The biological system is much bigger and stronger if we get it in the right shape. So improving the capture and storage are opportunities to reverse emissions into a negative scale. And you need a square megameter, like 100 million hectares, which is the size of our agricultural estate. This is the pasture, crop, and rangelands with some productivity in this country. And if we flip that into a regenerative situation, it can be, rather than emitting carbon, it can be drawing down carbon. And this gigaton challenge that Australia could deliver to the world would reverse our kind of current export of fossil energy as a liability into a real opportunity for the country. This is the same scale that was just reported by the Nature Conservancy for the United States, that with reforestation, which we're talking about, and natural forest management as the two dominant things, as well as improving agricultural landscapes, they can deliver a gigaton per year in the United States through these regenerative approaches. And the natural forest management is essentially trying to do prescribed fire and thinning of overgrown and sort of labile forests. So it's these applied sort of outcomes that can make a difference for food, ecosystems, and climate. And this is a big, probably 80-meter tree on the ANU campus, coastal campus, that it takes 11 of us to get our arms around. So this is real inspiring. Can we put millions of these big trees in the ground for the future? And this was a photograph I took when I went to visit Margaret. It's kind of this industrial ecosystem, or a rebrooding of the primary industry. It's going to take all the mining capabilities that we have with all the agricultural capabilities that we have to make a difference. So thanks very much, and I'm happy to take any questions.
Thanks, Justin. We have time for one question.
Carson.
So you were saying with Melliodora, the biggest drivers are, of course, temperature and rainfall. Is that taking into account soil types, microbiomes as well? Or
Oh, good point. So one of the higher resolution terms in that particular model was, I think, soil nitrogen at a certain depth. And so, yeah, Rose and I used to debate this a lot. Is it soils or is it climate? It's both. The answer is both. And the big climate drivers are not just simply temperature and rainfall, but exactly the seasonal extremes, which we just heard about. So there are different bioclim factors that come in, but I think the future is to actually get those crop models to try to look at the dynamic changes throughout the season and make the predictions directly. Yeah.
If you're successful in sort of kicking this off, what sort of annual investments required from Australia to actually...
$100 billion. $100 billion. That's 1 million farms. 100,000 a year to build it out, and that's the size of the current agricultural industry. We're at 60 billion, and they want to grow it to 100 billion already. That's a shift in the mining sector. So right now we have over 150 billion in the mining industry. Half of that is coal, which needs to be switched over to basalt. They could mine some climate drawdown rocks, and iron. There's another project to clean up the iron sector. So it's a structural shift within agriculture and half of the mining sector. So a lot of people say, oh, this is just impossible. This is a huge cost. It's actually an opportunity. It's economic structural readjustments on the scale that we've seen in the last 20 years. We've seen agricultural industrial agriculture rise all around the world. So this is the next stage.
I think we better continue this discussion. It's a very good topic for discussion.
You. So landscape, we'll get into the details of the landscape genomics in the workshop this afternoon too, so please join for that one. Thank you.
Thanks so much to Beck for asking me to chair this session. For those of you who I haven't met yet, my name's Rebecca Jordan, not to be confused with the lovely Rebecca Jones, who runs the organising committee. Both of us working in conservation genetics in this beautiful little city. It gets a little confusing at times. As with the other sessions, we have a really amazing lineup of speakers, and I'm really excited to hear the amazing research that's coming out. And so without further ado, I'm going to hand over to Paul Reiner from the Hawkesbury Institute for Environment, Western Sydney University.
Thanks, Beck.
So today I'm going to present what is an overview of some of the outcomes we've got from an ARC linkage. So this is a partnership with West Australia, Department of Conservation, Biodiversity Conservation and Attractions. And so what we've got here is really a roller coaster of what we've been doing over the last couple of years. As we know, climate change is impacting. In addition to the extreme events that have been documented to lead to mortality events here in the Jarrah Forest, there's also other stresses here documenting some pest and disease that are impacting in these systems. What we were looking at is a number of different questions going through looking at the genetic variation. Is there adaptive variance there? Trying to validate those by looking at quantitative genetic trials, and here we're looking at fitness-based traits and then functional traits that might be important for adaptation. So looking at our first question, we surveyed the landscape to try and capture as much of that geographic and climatic variation. We used the reduced representation DART-seq analysis. We started off with something like 40,000 SNPs, filtered down to 9,500, which we used to get an understanding of the variation in the system. So the first point of call was really to say, is there any genetic structure? And overall, there is, as Margaret presented in a number of different systems, widespread connectivity with a very low FST of around 0.05. The power of the markers allows you to have a look at some of the clustering within that, and we came up with 6 significant genetic clusters which are distributed throughout the landscape. But our main question was really about the adaptive variance. So using some of the tools, univariate analysis, we employed a number of different approaches, LFMM, Bay-EMV, Bay-PASS, and among others, essentially to try and see these patterns of correlation between climate of origin and the genetic variation in the system. Overall, what we were pushing was a hypothesis-driven, where we thought the extremes of temperature, the rainfall variation, and the aridity would actually be driving the distribution of these adaptive variants. So presenting those up front. What we wanted to do was have a look at those variants and then try and unpack whether those variants have any functional significance. So fortunately, there is a Corymbia giraffe genome that we were able to use, and we mapped onto that genome to be able to see if we had our variants inside the gene space, adjacent to it in the regulatory region, or outside of that. And so we could see whether some of the SNPs resulted in a change in amino acid. That's the non-synonymous in blue. And then the other region that we thought were interesting was the regulatory region, which can have a role in the expression of genes. We could map these across the genome. So this is actually showing the 11 different chromosomes of the Corymbia. And on the outside ring, we have temperature, precipitation, and aridity with each one of those outlier markers. Clearly, they're well distributed through the genome. Just highlighting one in particular. Let's see if I can work this. Yes. We have this little guy here, J8105, and this SNP in particular was interesting because it showed a very high turnover when we look at the change with climate. So just walking through this slide in particular, we have the non-synonymous changes, so that would lead to a change in amino acid and function. And here we have the regulatory regions for what is the max temperature, rainfall, and aridity. And we have the change, which is the turnover on the y-axis of these adaptive variants. So with that one SNP, we can see that there's a high turnover rate. So effectively, it's shifting from basically being fixed at one end to almost fixed at the other end for the different SNPs. So what we did from there was we looked at the variation across the system. Using a GDM analysis, we could actually predict where the variants were found across the landscape. And then from here, we can actually look into the functional annotation. So interesting, this is an aldehyde dehydrogenase gene, which is known to be important for abiotic stress, and in particular, heat stress. So that's one candidate that we can pursue further. But what we wanted to do was validate the variation that we have in the system. So using quantitative genetic trials, which have been demonstrated in Brad's talk, among others, to be important to have a look at what is fitness and potentially trait variation. So here we had trials designed by Richard Masnek in WA. There are 2 trial sites, both of them in the south. Here Margaret River is wetter than the Mount Barker site that we have over this side. Each of the trials has about 4,000 trees that we measured, height, basal diameter, and a blight resistance score. in summary, we get low to moderate levels of heritability for these traits, and then we looked at the QST, which is the differentiation of that trait among our populations. Comparing this to what is the FST, that is based on based on our SNP markers, we can see that there's significantly greater divergence in the trait, suggesting that they may be under selection. After doing a statistical analysis, we found that height and blight were significant, suggesting that they were the most likely candidates for selection. And then we looked at the relationship of those variants, the traits, with our climate of interest. So here we have height, and the other axis is blight, basically plotted against maximum temperatures. So across all of our plants, we can see that there is a correlation across this system, and we've been finding that correlation consistently across multiple years and multiple trials. So we're not finding a G by E interaction in this particular case. So what we did from here is develop a GLM model, and while we could throw in many climatic variables, which could predict 80% of the variation to predict across the landscape, what we decided to do was use our key climatic variables, those 3 that we started with, and in each of these models, 2 of them was able to explain a high percentage of the variation. So we used that because then we can interpret the outputs as we go to ground. So maximum temperature in both cases was able to predict the trait value in the system. So what we find from this output, we can actually go to the landscape and select particular plants where we know that those trait values may be present. So for example, in the southern area of the distribution, you have a very high growth rate. They have very tall trees relative to in the north, and as you go for the blight resistance, somewhat similar pattern, and you can choose plants that have high growth and high blight resistance, and within this landscape also unpack different combinations and be able to tease apart those different traits or combinations you'd like. But what we're going forward to do is intensive measures to try and pick out some functional traits that might underlie the different capacity to grow or indeed survive in these different environments. So here we use the same trials. Actually, we focused on Margaret River, and we took a subset of the trees here, studying about 800 trees, which are from 12 selected provenances. In fact, 4 of the provenances highlighted with the large circles were intensively sampled so we could get a greater estimate of the variance within those, and indeed estimates of heritability. So we did the growth measures, but we could use the differential growth. So we had October and then March having the integrated summer period of growth, and a range of leaf-based traits, specific leaf area, the dry mass content, and then the stem-based trait, wood density. And that was nice to see Brad's study pulling out wood density as a particular trait of interest. We went out there with spectral analysis, which pulls out a lot of information, but just came up with some nice, simple indices, and then used isotopes to try and get at water use efficiency. So this is the distribution of trait values that we have for all 4 of those traits I just mentioned. And for our point of view, just plotting this against the precipitation of the driest month, which pulls apart all the provenances so you can see if there's any clinal distributions. What we also estimated, again by having larger numbers for 4 of the provenances, is to estimate each one of these 4 traits, among others, the heritability values. And we do find moderate levels of heritability for some of those traits, but actually a non-significant value for specific leaf areas, suggesting that that trait in particular is very plastic. What we wanted to do is have a look at pairs of traits that we thought might be coordinated to each other and plot how those traits might be distributed across the landscape. So here we have delta C13, our water use efficiency trait, against the photo stress index that we have. So we estimate that across the current distribution, and then what we wanted to do is see how that might change into the future. So using the GAN model, we projected to the future, and then the key point here is what is the difference in the trait values that you might expect. And so this is the percentage change in those trait values across the landscape. So clearly some areas have high change relative to others. Moving forward in terms of the ability to adapt in the system, what we wanted to know is the traits that we think are coordinated, such as these 2 traits, are they actually changing at the same pace? And what we find is there is a parallel in terms of how they change. So we have the contemporary in blue, changed future environments in red, and those are parallel. However, what we see is that the one-to-one line, that would be the coordination that we see today, is actually shifting. So potentially they'll come into points where those trait values are no longer coordinated. However, when we have a look at these traits, which are associated with the leaf economic spectrum, that is specific leaf area and nitrogen, we find that there is total discoordination as we move into the future. So potentially one of these traits is actually having to constrain itself to match the other to have a more optimal phenotype. So just to summarise where we're up to, what we've got is a number of different studies, firstly looking at adaptive variants across the landscape. We do identify a number of different variants that could be associated with climate of origin, and indeed have mined down to see some of the variants and their functions, so we have some reasonable candidates in play. On the other side of the fence, using the quantitative genetic trials to try and see heritability of fitness traits, functional traits, and have a look at some of the coordinations that we have in play. In terms of our summaries, we find that, as predicted, height is actually adaptive. We have blight being adaptive, but in this case, the interesting story here is that that blight was introduced from the East Coast. So it's a new pathogen, a novel disease that's spreading throughout that landscape, impacting on the system. We predicted that actually, because they've never seen the blight, it would actually have no adaptation. So we have some pre-adaptation in that system, maybe because there's other pathogens that have a similar response. Our functional traits are indicative that we have traits that might be important for persistence, survival, maybe growth. What we wanted to do out of this project as a goal is really understand whether we have the variants to respond to climate and whether we could use that to inform some management, in particular assisted gene migration in the system. Given the low levels of differentiation across the whole system, the variants that we have, potentially we could move them into landscapes and maybe improve the viability of populations, but that's yet to be tested. Where we are now is we're taking those phenotyped plants and we're genome sequencing those. So with ANU, we're in the Borovitz lab, we're pushing forward with sequencing 480 individuals where we have that phenotype information at depth, and then we're going to look at the association between what is the genome and the traits, and then back to our climate to try and join this holy trinity that we try and push forward with. In terms of the other experiments, we've done lots of work in the glasshouse, the polytunnel, and most of which is looking at stress events. I'm just highlighting one experiment that we pushed forward to try and pull out this G by E interaction. We have provenances distributed across the landscape that we've pulled into the polytunnel, grown them for a long period of time under a sustained water environment of 100% availability or reduction. That reduction reduces growth, but they continue to grow and then it realizes new traits. And so we're interested to see if some of the traits, wood density, but also the hydraulic traits that determine the capacity to respond to drought are impacted. And some of the findings we're just showing down the bottom here are there's enhanced plasticity for populations that are from the arid, and indeed when we look at what is the predicted as well as the observed drought mortality in the system, we're finding that some of our mechanistic models are really playing forward. So we're trying to bring in that mechanistic component with it. This was a big team effort, particularly in the field campaign, and I think we had more fun in the field than a lot of other people. Went out there with a big team. At times there was a dozen of us playing around measuring our plants in Margaret River, not a bad place to play. And so I'd like to thank the partners at Murdoch, DBCA, and the grant that we got to source this funding. And I'll just leave on the last slide to say, stay tuned for some of the other talks. Colin Oren's going to talk about some RNA-seq, John Whale, and there's a poster up on the JARA that Jow has, so I think if you're up there at lunchtime or afternoon, check out that. We've got a landscape genomics workshop this afternoon, so we're going to explore some more of the comparisons that we can build forward. And if you're interested in PhDs or you know someone that's interested, there's some opportunities to come on board. Thanks.
Thanks, Paul. Any questions?
Justin.
Well, do you have any morphological traits with higher heritability than some of these phenotype than these physiological traits in your trial? Like some positive controls that you think will be able to identify the genetic basis of?
Yeah, so from the trials,
Yeah.
we do have blocks with replicates. So for the growth and blight, we were able to estimate what was something like 24 reps, but you still get that variance on the field. For the other traits, we had fewer reps, and we got those variants around the estimates.
50% or more heritability?
Yeah, not from the field trial, but when we take it to the glass house, then we have more controlled conditions, and we can pull out not the heritability in its narrow sense, but when you look at the broad sense heritability, we're getting sort of 40% heritability for some of the traits. Rose.
Are you able to control for the biogeographic history, the background genetics, when you're looking at those clients, relating the traits to the environment?
No, we haven't drawn the dots. No, indeed. So in our analysis, we basically just use the quantitative genetic traits and map those on, and then independently the genotype. So I guess we're still yet to join those components, and maybe some of the models that Dot was inferring could actually get us closer to that point. Yeah. Thanks.
Time for one last question.
I was wondering if you take into account the effect of epigenetics in the adaptation to climate, for example.
Yeah. I think it's fascinating, the epigenetic component, and it indeed could be a mechanism for adaptation. Because we haven't got clonal material and we haven't actually pushed it even in our experimental manipulations at the family level, we haven't really controlled for that level of genetic background. But we have done those analyses based on the population. So what we're estimating is the plasticity. We haven't actually gone further to quantify methylation or other levels of epigenetic signature that would be in play. We have done some RNA sequencing, which would be the expression of those patterns, but not the methylation per se.
Great. If you'd all like to just join me in thanking Bob. So our next speaker that we have up is Kevin Murray from Australian National University.
Good everyone, thanks. So I'm a PhD in, just recently submitted my PhD in Justin's lab at ANU, and today I'll be talking about some work I've done as a collaboration with Rose Andrews' lab, looking at landscape drivers of genetic diversity in woodland and eucalypts. And so like a lot of the studies in this session and in landscape genomics, we're really interested in interrogating spatial genetic patterns. In particular, we're looking in wild individuals in their natural environments, and hoping to perhaps uncover some of the patterns that have been left by evolution to various environments. And so for this study, the specific patterns we're talking about are whole genome average sort of spatial patterns, like discrete population structure, where you have quite distinct groupings of individuals, or isolation by distance, where genetic differentiation is more gradual like the landscape, as well as isolation by environment, which is where similar environments, individuals in similar environments are more genetically similar than you'd expect just by their geographic proximity. There are many processes underlying these patterns, both neutral and adaptive, and for this study we're completely agnostic as to what the underlying causes of these patterns are. The system in which we're asking these questions and studying these patterns are two species of woodland eucalypt, Eucalyptus albans, white box, and Eucalyptus soteroxylon, mugger ironbark. They occur from sort of western and central Victoria through southeast Queensland, broadly on and northwest of the Great Dividing Range in southeast Australia. They have overlapping but distinct geographic and environmental ranges, and taxonomically they are in different series within Adaptation, within the Symphomertens. The aims of this study are essentially it's a descriptive study where we're aiming to characterise patterns, to determine the genetic diversity and differentiation between species and localities, describe the relative extent of discrete and continuous population structure, isolation by distance, and isolation by environment. And this is going to lead into future studies where we're actually doing genome scans for genotype environment association as well as differentiation between species. And so the approach that we've taken to do these studies is a sort of a cost-conscious whole genome sequencing approach. The molecular work for which has been spearheaded by Ash Jones and Norman Vathman in Justin's lab. On roughly 200 individuals of both Eucalyptus albans and Eucalyptus cytoroxylon from 20 to 25 localities from southeastern Australia. Overall, per individual, we have somewhere in the order of 5 to 10-fold coverage. And in total, across all of the various bits of sequencing that have been done, that totals over 3 terabases of data. So it's a quite large data set. So what do we do with that data? First, we align to the Eucalyptus grandis reference genome using a combination of both hard-called variants with freebase and an angst-based pipeline, which Rose mentioned yesterday. So obviously this is not high coverage data. There are various errors in, and lack of confidence in the genotype calls, and being able to carry through that uncertainty further into analysis than just the genotype calling really improves the estimation of various population statistics. And that's been done within angst. Going on from that, using construct, which is a new method for jointly modelling both discrete and continuous genetic differentiation. Both Paul and Margaret this morning showed what happens if you have a nice isolation by distance clone and you feed it into something like structure. You get out those sort of clonal grades. Construct takes a sort of different view on that, which is you allow for both things, both continuous and discrete processes, and then through model selection can quantify them relatively. As well as generalised dissimilarity modelling to sort of tease apart the isolation by distance and isolation by environment across the landscape. And that GitHub logo is there to remind me to say that a lot of work went into the engineering behind this, and it would be a bit of a waste if we only used that for one study. So all of this is generalised, it should be applicable to other studies, and it's up publicly available, and I'm more than happy to help people use it for their studies if people are interested. So what did we actually find? Quite high genetic diversity, as everyone who looks at Eucalypt genetic diversity seems to find. In our case, average pairwise distances somewhere in the order of 5%. Across the genome, we found 130 million SNPs, of which 70 million were in at least 10% of samples, or the minor allele was in at least 10% of samples. As well as 55 million, or of that, 55 million were had minor allele frequencies greater than 10% in both species. So quite a large shared gene pool. Very short linkage disequilibrium. Well, we estimate a very short linkage disequilibrium in the hundreds of base pairs, depending on varying across the genome, obviously, and very low divergence between localities, somewhere in the 2 to 3% FSTs. And around about 15% FST between Eucalyptus albans and Eucalyptus cytoroxylon. When we looked at population structure using construct, we found no support for strong discrete population structure and support for isolation by distance. And unfortunately, that doesn't look very good in a structure-style bar plot because it's all just one colour. So instead, here are some PCAs. So on the left, Eucalyptus albans, and you can see exactly that pattern. There's no strong discrete grouping here. The leading principal component axes explain very little of the overall genetic variation. In Eucalyptus cytoroxylon, same, although the axes here explain a little more of the overall genetic variation. When we dig into isolation by distance using generalised dissimilarity modelling, which is a non-linear way of estimating or predicting genetic distance with a combination, well, in this case with just geographic distance, we find that Eucalyptus albans has moderate correlation between genetic and geographic distances in an approximately linear fashion, with an overall variance explained of around 26%. Whereas in Eucalyptus cytoroxylon, the correlation between geographic and genetic distances seems a little quite strong, somewhere in the order of nearly 80%, and in this nonlinear pattern where at stronger divergence at greater distances. When we look at isolation by environment in Eucalyptus albans, so this is going through a variable selection process where you first included geography in the GDM model, then you successively add environmental variables that explain further variation, and then you use the backward selection within GDM to weed out ones that aren't significant. What happens at the end of that is you see things like precipitation seasonality and total amount in the wettest quarter, soil water conductivity and soil phosphate concentrations being important. And overall, across all of those variables, that explains an additional roughly 6% of genetic distances. And on the right there is just a map of the relevant area, coloured by a composite of all those environmental variables. We do the same thing in Eucalyptus cytoroxylin. The variables that pop out are more around humidity, the rates of change in both temperature and humidity through the year, and cloud-adjusted solar radiation. And overall, across those variables, that explains an additional 12%, bringing it up to a whopping 90% of genetic distance that can be explained by a combination of geographic and environmental distances. So what are the biological implications of all of this? These species have very large, mostly continuous distributions. They, as you'd expect from that, have very large shared gene pools. The diversity and estimates of isolation by distance are on par with similar studies and previous studies in similar species, closely related, particularly Jordan and Megan Supple and many, many others. And that joint inference of isolation by distance and discrete population structure really helped us make a clear picture of what was going on. Isolation by environment does somewhat correspond to the species niches. Obviously, big flashing warning, correlation, not causation, but Eucalyptus albans seems, according to where it grows and in textbooks, to prefer fertile, well-draining soils, and things like soil phosphate and soil hydrological conductivity come up as important. Eucalyptus cytoroxylin, on the other hand, extends more into arid areas of Australia, and things like moisture demand and humidity and radiation and the overall amount of rainfall come up as important there. So from first principles, that makes sense, and these are the variables that everyone seems to select whenever they do these studies in eucalypts. And in terms of conservation, these species have huge adaptive potential, and let's focus on preserving that with methods like climate-adjusted provenancing that several people have mentioned so far this morning. So in future, moving forward beyond my PhD, I'll be hopefully focusing on doing genome scans for whole genome, using this large whole genome data set to look at both genotype-environment association and looking at how variation or how differentiation between these two species varies over the genome. However, that could come with some challenges just because of the huge diversity of these species. So with that, thanks to everyone who helped make this study. My co-authors, Jasmine, Helen, Ash, Rose and Justin, various other people who assisted in many ways, the people who supervised and funded this. And with that, I'd like to take some questions.
Any questions for Ken?
Why is so much of the variation in genetic distance explained for
Very good question. So for Eucalyptus albans, it seems to be roughly on par with previous reports in, say, Melliodora. So in terms of the differences in technology and the approach, which are quite different to what some other people have done, I don't know that it's necessarily a technical cause. We kind of had a couple of hypotheses about why cytoroxylin could be lower than albans around pollinator ecology, but it's something we'll dig into. I believe although how far into the past this was the case, I'm unsure, but I believe it also has a patchier distribution, and therefore that could obviously reduce gene flow over larger distances. But yeah, yes, some combination of all of those. I mean, differences in flowering time, range as well.
Haven't you also tried putting the albins and the cider together?
Yes.
Given that 50 million SNPs are shared between them, do you want to tell them what the results are in the joint analysis?
The joint structure analysis?
Isolation by distance and environment with and construct with the joint analysis?
Basically that they fall out as two, well, if you treat them as populations, they fall out as two populations with relatively different isolation by distance curves. I mean, just as they do when you treat them separately. And yeah, haven't done the isolation by environment stuff on that yet. That's next week.
Any more questions? Okay, we'll try to get time for one more, maybe two short ones. No, we might wrap it up there. Thanks so much, Kevin. Institute of Environment, Western Sydney University.
Awesome, thank you. Well, when I submitted this abstract, I got really excited, and I submitted this awesome title that so I was going to show you two different experiments. It turns out I have way too much data for that. So this is almost a lie, and I'm only going to show you one, but I have one slide about water manipulations at the end, so it's not a lie. So, moving on. So I think the yesterday, today, and tomorrow are really showing us 100 different reasons why it's really exciting to study eucalypts. There's so many different reasons. They're widespread. There's lots of them. They're really old. They're economically important, etc. And some of the reasons that I think they're really neat to study is because they're widespread and they have overlapping distributions. And that means we can ask questions about evolution and adaptation among different species. Now, at Western Sydney University, there's a group of physiologists whom I don't consort with, so don't worry. And they're doing some really nice work though, and they're studying Eucalypts tereticornis, grandis, and all this other stuff, and they're looking at how water and temperature might affect general physiological adaptations or phenotypes. So I'm going to walk you through. I'm going to walk you through some of the basics of why they're finding what they're finding and what I'm interested in, and try to marry those two together. And so if we take Eucalypts tereticornis that's present on eastern Australia right here, and you have two distinct populations, one in the temperate region and one in the tropic region. And if you throw one in some individuals from the temperate region into its home environment, let's just call this 21.5 degrees Celsius, and you can measure any sort of phenotype. And it could be photosynthesis, it could be growth, it could be anything like that. Now, if we grow the same individuals in a tropic type environment, 28.5 degrees, it's pretty unhappy here. And so when they measure these phenotypes, they can measure one. Can take one measurement and compare that across. However, there's lots of other stuff, underlying mechanisms that make that phenotype happen that they're not able to measure. And so that's where I'm interested in. What are those mechanisms? Are those mechanisms the same within a species across different genotypes, from the tropic to the temperate, and are they the same or different across species? And so you might get some output like this that shows genotype by environment interaction. And hopefully it's fun to get that because it's interesting and it's publishable. Now, the two species that I'm interested in are Eucalyptus grandis and Eucalyptus tereticornis. And so, and for a lot of the same reasons I mentioned before, is they have overlapping distributions, and they're widespread, and so they traverse many environmental gradients. And so I've got a couple dots up here of the populations that were collected. So we have a couple temperates and a couple tropics, and we're going to grow those and we're going to ask a couple questions. So I have two basic categories of questions: within species questions and among species questions. And the within species questions, are there going to be similar differential expression among genotypes? And is there going to be an environmental signature? Is there going to be a genotypic signature or genotype by environment signature? Now, among species, I think this is where I start to get excited, where you can start looking at orthologs and see if those orthologs have changed or remain the same in terms of expression. And so hopefully I'll walk you through this experiment, and you'll be as convinced as I am. So quick methods slide. So two growing temperatures, 21.5 and 28.5 degrees Celsius, 4 different glasshouse bays, and so we had replication among glasshouse bays. We had 2 different plant genotypes per species, temperate and tropic. When we collected the samples in the middle of the day, and we snap froze them in liquid nitrogen, stored them in -80 freezer, and we ran RNA-seq on them. And here's some information. We used Eucalypt grandis as the reference transcriptome, and got about 16,000 genes. Our mapping rate was between 70 and 80% for our RNA-seq data, which is pretty decent. And here's some pictures. So Tereticornis is on the left and grandis is on the right. You can see there's variable phenotypes in there. And so I want to see what's going on underneath all those phenotypes. But we did measure some phenotypes, and so we can compare what's happening with differential expression and the phenotypes. And so this is the only phenotyping slide I'm going to show you, and it shows pretty well. It's just growth, basically. We have biomass on top and leaf biomass on the bottom, grandis on the left and Tereticornis on the right. Basically, Tereticornis shows really nice G by E patterns, whereas grandis doesn't. This is more phenotypic plasticity and or genotype signature. So here's the experiment. Well, this is more the statistical analysis. And so basically, here's my 2 genotypes, my 2 environments, and some happy and unhappy phenotypes. And if we make contrast between the environment, we can think of this as expression differential, or expression plasticity. So phenotypic plasticity, if we just compare everything grown in 21 degrees versus everything compared to everything in 28 degrees. We can look at the genotype, so we can make the opposite contrast. So we can compare all the temperates grown in both environments versus all the tropics grown in both environments. Lastly, we can compare we can figure out if there's genotype by environment signature by comparing the two homes, the 2 happy trees versus the 2 unhappy trees. So now, when we get to 16,000 genes, we can get a general look at what's happening. And so this is just a multi-dimensional scaling plot. And this shows for both species, this shows that there's just a really nice environmental signature and not much else. There's probably a little bit of grouping of genotypes, but that environmental signature is pretty big for both, and so that's pretty interesting. So let's look at that first. This expression plasticity is what I'm calling it today. Tomorrow might be something different. So we have this is a heap maps, and each column is an individual, and each row is a gene. And the dendrogram on either side, it shows you how closely related those individuals or genes are to one another. And they come out as nice groups here with the environmental signature. And there's some overlapping in genes as well. So in total, there's 283 genes we found significant in Eucalypt grandis, and about 200 genes we found significant in Tereticornis. And those 61 genes that we found significant in both species. Now, if we think about those 61 genes, we can look at them and look at the log fold change, which is the y-axis here. So this is the differential expression of the genes within both species. So each color is considered a different species. And you can see here that 60 of those 61 genes are actually showing the same pattern among the environments, except for this one stinky little gene. I don't know what's going on there. I'll have to think about that. But that's pretty interesting. So this differential expression patterns it has been conserved across species. So if you look at those, so this is my gene ontology analysis. This is just a MAP-MEM output. And there's lots of interesting stuff going on here, but I'm just going to show you the heat shock proteins, which are down here. So there's lots of heat shock proteins that are involved in both species, more so in Tridocornis when you think about the environment, less so in E. grandis. So now that we've got a picture of what's going on in the expression plasticity arena, let's look at the genotype effect. So if we look at the genotype effect, we see a pretty similar pattern. All the temperate are on the left, all the tropics on the right on both. And we get nice patterns. They're all the dendrogram shows that they're related to one another, and the genes are pretty similar in terms of total genes per species, but the number of overlapping genes goes down. And so when we look at that and in terms of species compared to species, we get exactly half. 10 of those genes have the same pattern, and 10 of the other genes have a completely opposite pattern. And so this I like to think about as somehow they've adapted differently to those the genotypes have adapted differently to those environments. So if we look at those heat shock proteins, now there's a bunch of heat shock proteins that are involved in E. grandis, but there's none in Tridocornis. So that's really interesting. So you've got this really nice pattern of plasticity, but no genotypic pattern in Tridocornis. So that's 5 minutes. And so I think I'm almost done. I should move along. So we got this really interesting pattern of plasticity, but no genotypic pattern in Tridocornis. If we look at the genotype by environment interaction, it starts to get a bit messy, but there's still a bunch of genes that are involved in this process. But what's really interesting is only 5 genes that are in common between the species. So if we look at those 5 genes, all these 5 genes show completely different differential expression patterns. And so this might be really important in terms of adaptation for each species, that they've found different ways to become adapted to the environment they're being grown in. So again, you look at the heat shock proteins. There's only 2 heat shock proteins that were found in Eucalyptus grandis, and none in Tridocornis. So again, pretty interesting, I think. So just a little conclusion slide. So phenotypic plasticity plays a major role in both species, and among species, it's pretty common that there's been a lot of it's pretty similar among species, so it's been conserved. There are significant differences between genotype within each species. And among species, there's actually there's some similar patterns, but there's some different patterns. So there's pretty interesting stuff going on here. So we can partially accept that hypothesis. Patterns of local adaptation are present in both species, and there's many more genes involved in Eucalyptus grandis and Eucalyptus tridocornis. And there's only a few common genes, and they all show different patterns of differential expression, which could be important in terms of managing those species further on down the track when climate change comes through. And here's my one water manipulation slide. So basically, in how much time do we have? 2 minutes? 3 minutes? So what do you guys think? Questions, or do you want me to talk about this? All right. So there's 3 times. There's time 1 where it's well watered. Time 2, we grow the plants in drought for a few weeks, and then time 3, recovery. And this right here is the same heat map type pattern, but time 1 are these 3 individuals on the left, time 2 in the middle, and time 3 on the right. And it shows a really nice pattern of differential expression between time 1 and time 2. So that's sort of a resistance or resilience to drought. And then those same genes are being recovered afterwards. And there's about 1,000 genes involved in this process, so there's quite a bit. And there's quite a few genes that are also involved in just time 1 and time 2, but then don't recover. And the opposite is true, that there's no change between time 1 and time 2, but then then are part of the recovery process. So that's quite interesting too. If you have any questions, we can talk about it. I got heaps of this data on my computer, so if you want to have a chat about it, happy to have a chat. With that, I'd like to thank everyone for coming out, and it's been awesome so far, so done. Any questions?
We have time for one super quick question. Colin.
No,
I think they all loved your slide.
That's not going to be quick.
Oh, sorry.
Very quick. You said that you see G by E, or just the genetic effects that they're adaptive. How come you wouldn't say that they're neutral? Why is it that?
That's actually a good question. And it's something that I've been playing a lot with in the statistics, because I was wondering if there were genes that actually were neutral at some point, but then were neutral for one species but adaptive for the other. And it gets even messier. And that's something I'm actually trying to work with, but I don't have a really good answer for that at the moment. Yeah, I wanted to be. That's right.
Thanks very much, Colin. Cheers.
Yeah, thank you. Do this?
So next speaker is Maya O'Neill from the University of Victoria in South Africa. Welcome, Maya.
Thank you. Good morning. I'm presenting a small section from my PhD project. I feel a little bit like Frodo from the Shire among all the big guns here. So I hope you enjoy. The PhD is on the population genomics of Eucalyptus grandis. Now it might seem a bit audacious for a South African to come here and tell Australians what's happening in their backyards. But the truth is South African forestry loves grandis. Our love affair with the species has been going on for more than 100 years. First for mining timber and later for pulping. We have stuck together through sickness and health. In the future, we are also worried about these same scary predictions. And South Africa specifically is predicted to become drier, hotter, and as with many other places in the world, have more extreme weather events in both severity and frequency. How that will affect production and how the plant interacts with the microbes and the other organisms around it is very hard to predict. And with all that uncertainty, it's more important than ever to have genetic diversity readily available to respond to whatever the challenges are and to get better at predicting which genotypes are going to work well for those environments. Lucky for us, in order to find matches for future conditions, in Australia, in the native range, there are provenances already experiencing the conditions that's predicted for our areas. And we can use that landscape genomics premise. I adapted this from a review by some people that are sitting here in the audience. Using that premise, we hope to take these principles and generate models for genotype by SAT matching. So with that, we wanted to determine and compare the neutral and adaptive population structure of native grandis. And I'm going to dive right into what we used for that. So over here, the yellow one, is that the laser? Yes. Okay, so we sampled all the way from the north in Windsor to the south in Willadella. If I don't say the place names right, please forgive me and correct me privately afterwards. Please. And you can see the sample size indicated by those circles. And there, it looks like a crazy person. I should be in a mental asylum, but I was absolutely I was drunk with power because in my hand I held a species. That was the day we got all the seeds that we got from the Australian Tree Seed Centre and from Professor David Lee. I don't know, I saw him somewhere. From Professor David Lee's private collection. We germinated these, collected DNA from leaves, and did the 60K SNP chip genotyping on those. And all these seedlings have now been put in a plantation and a trial in KwaZulu-Natal in South Africa to serve as a long-term gene conservation bank. So in total, we have 194 families represented from 33 provenances. And although we genotyped just under 600 samples, to keep it balanced so that no family is overrepresented, I'm working with a 362 individual sample set, and we have about 26,000 informative SNPs. The neutral or the demographic history that we got from PCAs and admixture analysis agrees very well with the geographic distribution, with one exception that these provenances in the Yangala area, they are more differentiated from the north and the south than the north are from the south, based on FSC values and also the admixture analysis. I can't go into too much detail because of the time constraint, but when we did a PC-ADAPT scan, we found evidence of integration in that area, which is why it has this differentiated value. Instead, I'm going to talk about how we're using environmental association analysis to help us elucidate the adaptive population structure. So just briefly, I know I don't have to introduce it to this audience, but just briefly, if we take elevation as an example, environmental variable, so here you have the provenances from the northernmost to southernmost point, and the elevation values. And say we have a marker in the genome where the 1 allele, the A allele, occurs at a higher frequency in populations with high elevation, we can say that that marker is in fact associated with the environmental climate. So that's exactly what I did. And I show here a Manhattan plot, and there is the elevation, markers associated with elevation in the northern subpopulation. And each purple dot is a SNP marker that had a significant association. So that is at a, it had a Q value, and it was a P value that's been adjusted for false discovery rate under 0.05. And on the bottom here, now it looks like a lot of purple dots, but on the bottom here, there's a whole lawn, a grey lawn of markers that had no association. So those are, in other words, not associated. And we use LEA, which is the LFMM, it's a latent factor mix model. R package. All right, so we wanted to see whether we get a different population structure if we use those grey markers, the putatively neutral, and the purple markers which are associated with elevation. And I'm just going to focus on one area where this was quite obvious, and that's in Paluma. So there we have two provenances that are about 50K apart as the crow flies, but they have very different elevation values. And if we look at the grey markers, the ones that weren't associated, we find that we can't differentiate between these two provenances, but obviously with the elevation-associated markers, we can clearly differentiate between the two. Now, we're not just serving our forestry community, we're also academics, so the first question that pops up is, what are the genes doing that's near these SNPs? So we found genes that are in algae with the markers, and did gene ontology enrichment for biological processes. Now, always compared with the neutral. So the neutral ones had biological processes that are very much central to cellular biology, compared to the adaptive markers, which were like responding, genes that seem to be involved in response, and phosphorylation and cell communication. So that's just elevation that I showed. There are other geological, environmental factors, temperature, precipitation, of course, soil factors. And if I have data that doesn't seem correct, you're welcome to blame your own wonderful resource, because I got it all from there. And because these are often highly correlated with latitude, for the other environmental factors, I use a principal component analysis and then do the associations with the principal components. So to conclude, that was a record. We have begun to elucidate the population structure of the species that is so dear to our hearts. This genetic variation is now available to South African breeders, which I think is very valuable. And of course, this is the first little baby steps. People are still holding our hands, baby steps, to start building these models. And of course, we have to finish that loop so that we can get the full benefit and start testing our hypotheses that come from this study. And in that, embedded within our study population of 193 families, are 107 families that CAMCOR, via their members, are going to plant in provenance trials in different parts of the world. And that is going to be the first common garden material that's correlated with our material. So thank you to the organisers for giving me a chance to present on my humble PhD project. I am regretting that that thing hasn't gone because it means I have 5 minutes of questions. So please be kind. I am but a student.
As Maya has mentioned, plenty of times for questions. So
fire away.
Predictably, from me, do you know what the other parent might be for the introgressed population? How much of the genome looks like it's introgressed?
So it's hard to say how much of the genome because there could be, we don't have whole genome coverage. We have, and these markers aren't in LD, but there seems to be a big region on chromosome 3 and 5. So big regions and then spotty regions along the rest. We have no idea what the other species is. I wouldn't even begin to speculate. We don't have access to those other species' genetic material. We only have the commercial species grown in South Africa. So if you have suggestions or you want to chat, I'll be very interested.
Sorry, this question.
All right.
Can you say anything about the origin of the most highly productive grandis populations that you have? I know there's a sort of a pedigree, there's information that previous have about what they think the population combinations have been, but can you add anything new to?
Yes, that is a very good question. In fact, that is chapter 2 of my PhD. So I will give you better answers next time we talk. But so it's very difficult to say because the material that was first brought into South Africa, they didn't have provenance information. It was kind of just seed collected here, sent to the Cape Colony, and that's where it came from. But there have been secondary introductions from Atherton and Coffs Harbour, which I think is also the provenances that popped up just before now. So there are those. What we are seeing is that they are mixing. So I think a lot of times we're selecting for intraspecific hybrids, so from that have crossed now between there. But what we also suspect we will find, and I think we're seeing some first evidence of that, is in South Africa we're growing them around Europhila and things like that don't necessarily have the reproductive isolation barriers because they didn't occur in Australia. So we suspect that we'll also find hybridizing with other species in the South African breeding program. So it's very difficult to say which provenance has been performing really well because they're all kind of mixing. So probably various kinds of hybrids, I suspect, intraspecific hybrids and some external stuff. And a lot of when Grandis was first brought into South Africa, they thought it was Seligna, they called it Seligna. In fact, Grandis wood is still called Seligna wood in South Africa. And so there might be that in between also. So it's very hard for me to say, but we'll do the genomics and then I'll get back to you. Paul?
You presented the elevation and then you showed a range of different other metrics. Can you say which one you think is more important, or which one is potentially showing the greater number of adaptive variants?
So it's hard to say which one because they are so correlated along that latitudinal range. So it's it's difficult for us to say it's temperature versus precipitation because they're correlated. They vary the same. And also the things that show more. It's hard to say whether it's more markers. Does that mean it's more important? It's not the strength of the. So it's hard to say. What I wish we could do is something that we saw Dan Jacobson present a few months ago while he was visiting us from ORNL. He showed that they want to take not just temperature average over the year or precipitation average over the year, but every day and draw associations with that because sometimes the association is about the shift from one season to the next. So that shift can be some organisms adapt better to that or respond better to that. So sometimes that's very hard to measure with average values. So I think we're not even scratching the surface in terms of what these organisms are adapted to. We need lots more computers and lots more data. But so I can't really tell you. I really don't know. Yeah, we're probably going to have to. Yeah.
Time for a couple more questions.
Xander, this counts as a stage gate.
How are you accounting for the geography along with the environmental variables? Because they're all correlated with each other.
Yeah. You mean in terms of the latitudinal range? Or what do you mean?
LEA put it all in?
Yeah, so that's a latent factor mixed model. It basically, the correction it does is for the overall population structure. So that's the correction it does. In order to correct for that, so that's basically the correction. It's that underlying population structure.
When you're trying to distinguish which environmental variable is more important, they're all correlated with each other.
Yeah, so then it's done. That is unfortunately not the 15-minute, is it? Anyway, so then we're correlating it with the principal component. So that's why I can't untangle which one it is. And it's not the same if you take the northern subpopulation and the southern subpopulation, because the environmental variables correlate differently in the south than they do in the north. So I have to treat those separately. Yeah, no, it was really nice. I enjoyed your talk. And yours. And yours, and yours.
All right. On that, can we thank Maya?
Thank
for a
you.
great talk.
Thank you.
Beautiful. So up next, we have another contribution from Paul's group, so from the Hawkesbury Institute of Environment, Western Sydney University. I should be getting good at saying that now. We've got John Brown.
Hi, thank you. I'm really excited to talk about my first part of my PhD with you today. And I will just start with a bit of background. Climate change is occurring. There are species and trees around the world that are dying. And so there are some species that have a capacity to adapt to this to a changing climate. But then there are also populations within species that do and don't have this outstanding capacity. So this is where I come in, looking at some of the red gums of Australia. If you do a nice Google search, you find that the red gums look like this, but they actually look like this. And so they're both widespread species. They're found on the east coast. Pterodocornis hugs the coastline, and Blakeleyi is on the west of the Dividing Range. So then this drives my hypothesis that Blakeleyi will have a greater adaptive capacity to the more climatic extremes because it experiences less rainfall. And therefore, rainfall itself will also be one of the main drivers for adaptation. But because they both have a latitudinal gradient, temperature, I believe, will be, the SNPs associated with temperature will be more shared across both species. So sampling the environmental, the geographic space is just not enough. We need to ensure that we sample the climatic variability as well. So just showing here maximum temperature with precipitation of the driest month, you can see that they both share that same, a very similar maximum temperature, but there's a bit of more variability with Blakeleyi in the driest month precipitation. So this drove my field collections. Leaf material went for DartSeek, and very hot off the press, it generated 61,000 SNPs, and through a number of fielding processes, this drove it down to about 7,000 for each species. From here, we've gone on to do some data analyses, looking at genetic structure, some environmental association analyses, which I'll focus on today, and then mapping these onto, or seeing where these fit on the grandest genome. So the first climate association analysis that I did was a multivariate analysis, RDA, with a number of different climatic variables, and between the two species, this generated 94 and 74 candidate SNPs each. When we compare this to LFMM, which is a univariate analysis, you see that we get much higher SNP associations with this, but also you can also see that there's a number of consensus genes, or SNPs, sorry, found between both models. Next, I wanted to look at whether there was any SNPs shared between the two species. So for precipitation of the warmest quarter, see that there's a much greater number of SNPs associated with this environmental factor for Blakeleyi than there is for Pteridcornis, and there's a very low number that are shared between the two. Inversely, when we look at mean annual temperature, Pteridcornis has a much greater temperature association here, but there's still a very low number of SNPs that are shared between the two species. Then doing a blast, looking at whether these SNPs come into any functionality or association with stress, we see that there's a heat shock protein and a protein that comes in with response to ABA. So these are like really cool, something I want to look into further. But then this brings me back to my hypotheses that Blakeleyi does in fact have a lot more SNPs that are associated with the climatic extremes, whether it's maximum temperature or dry precipitation, that rainfall itself has a much greater influence on the Blakeleyi associations and the adaptive variants that you see in there. But then when I look at for the number of SNPs that are shared, I don't know yet whether there's enough with temperature that gives, and more analyses need to be done on this. And so I would like to thank all these people who have fallen into rivers doing fieldwork with me, and then funding bodies. Thank you.
One quick question for John. Yeah?
No?
No?
Good.
That's all right. I'm sure you can catch John at lunch and fire away some more. So thank you very much. Okay, so our next speaker is Soil.
Yeah, sorry. Yeah.
Soil, fantastic, who's from the University of Canterbury, New Zealand. Thanks.
Yeah. Hello, I'm Sir Jong Kim from University of Canterbury, New Zealand. I'm going to talk about population, genomic, and taxonomic study of Eucalyptus agriflorea and Bosistwana. So my PhD project is funded by NGDFI. I needed to talk a little bit about the background. The NGDFI is commercially oriented research and development project, which aims to create plantations of high-value, durable eucalyptus timber species in dryland environment. So there are reasons for growing the durable eucalyptus. It's because hard, strong, and naturally resistant to decay, so can be used without any chemical treatment, such as CCA. And there is high market demand, just one example. So timbers are easily broken during the mechanical harvesting in vineyards, which probably make the wine more expensive that we are not so happy about. And durable eucalyptus can be a good alternative for that, and there is already a good business going. Also, it has a good high potential to grow well in dryland in New Zealand, proven by demonstration trials in New Zealand with the selected species. So NGDFI collected seeds from mother trees in Australia that has a good looking, and each tree forms a family in which we know the mother tree but not the father. And the seeds collected were planted in trial sites in dry land around New Zealand. That looks a bit like a cemetery, but actually trial. Yeah. So the best performing tree species were E. Bosistwana, based on measuring various wood properties. So NGDFI was happy to continue the progeny test with Bosistwana, but some strange things were found. In this picture, these two differently shaped leaves are from the different tree, different branch, but actually from the same family group, and it's supposed to be R. Bosistwana, but it looks so different. I personally don't know which leaves are from what species, but some NGDFI people reckon that one of them has the same trait of the E. agriflorea. So the problem is this morphological confusion happens in so many trials in New Zealand now, and no one is now sure about if it is agriflorea or Bosistwana. So based on these uncertainties, we got 3 research questions. First, what is the taxonomic identity of the morphologically deviating populations of E. Bosistwana? If it was misidentified or mislabeled from the seed collection, or if the mother tree was already a hybrid origin, or it was hybridized during the progeny test. So, or if the taxonomic delimitation of the Agrofloria and the Bosistona was unsolved yet, as it is closely related. If in that case, we will look further into this. And second, what's the population genetic structure of the E. Bosistona? We are wondering how many genetic populations are there in the New Zealand triads, and even beyond that from the mother trees in Australia. And also, what are the patterns of genetic connectivity and the structure of the population? Last question, what's the genetic structure within families of E. Bosistona if offspring from the same mother trees are half siblings? As you know, some Eucalyptus species are notoriously promiscuous, if it is correctly pronounced. And there are sometimes mix of the family relationships among the progeny. And to solve this, we'd like to reconstruct the pedigree to better inform the breeding program. So I just started my PhD last October, and I got only PhD today, but we are anyway underway. So we are participating in the 65K SNP array initiative, and to generate the SNP data to solve those questions. And working on a lot of DNA extractions, and it's not working excellent now. So I'm working with a Quaisen plant mini kit, but I tried several modifications, but it's not working perfect. So I would appreciate if you could share the protocol with me, or can talk later in person. So I'm quite new and not included in the group picture yet. My group UC Concert, so I added my own picture there. Thank you.
Shout out to all of those who have done DNA extractions. I'm sure storage will be. There you go.
Yeah.
Right. Thanks very much.
Thanks.
So we'll move on to our final speaker for this morning's session. Beautiful. Oh, just like you've got a pocket or a hook on my pants. Yeah,
Hook it on my pants.
So our final speaker this morning is Eleanor Fox from Federation University of Australia.
If I can get the microphone on. I'll just hold it maybe.
No worries.
So my name's Elle Fox. I'm presenting my PhD research so far. I have a bit of a confession to make. I actually did my honours in herpetology, and then I went from there straight into a land care job for 7 years, which is where this project kind of was born out of. Because after 7 years, it's a long time to start asking, what am I planting and where, and is that any good? So, excuse my notes, I tend to go off on tangents. So, we all know over the past 200 years, land use in Victoria has drastically changed, and the landscape has been significantly modified and really quickly as well. So, the habitat destruction and clearing of vegetation has resulted in fragmentation of plant and animal populations, and changes in ecological processes such as the pollination regimes of flora. And this has led on to the change of ecological functions of those sorts of things. So these effects are especially prevalent in southwest Victoria, or the agricultural hub, and in eastern Victoria, where mines and power plants have gone in and are now in the process of becoming spent or closing down or moving on. So many trees in these areas that used to have bigger populations now reside mainly on roadsides, especially with remnant members of those populations, and continue on as paddock trees as well on private properties. So there's recently been an increase of restoration, and by recently I mean like past 30 years, so relatively recently, with mines and power plants and logging companies now legally required to rehabilitate sites after they've been spent or decommissioned. So this is a picture here of Hazelwood. That's over in eastern Victoria. I'll tell you a bit more about that later, but it's been shut down now, and so now they're looking at rehabilitation. Also, there's been a change of farming practices over those past 30 years as well. So when shelter belts and re-veg projects first started out, it was non-native species, you know, your pine tree, cypress, things like that. Then we had another change into Australian natives, but it was just pretty much any Australian native. And so now we have a lot of Western Australian swamp mallets and things like that in Victoria, which is doing okay. And now we're kind of moved into local provenance. But with upcoming climate change and things like that, is this the right thing to be doing still? So when organisations such as Landcare run tree planting projects, we're covered by insurance for a 30% death rate, but often it's a lot higher than that, even if we're planting local provenance. So why is that happening? Why is there even a 30%, let alone sometimes a 50, 60% death rate in these plants that we're planting? And so does that mean that we're still planting them in unsuitable areas even though it's still local provenance? And so is the genetic diversity present in these projects to ensure ongoing survival of these species with climate change? So I'm looking at two species. I'm looking at yarragum, Eucalyptus yarriensis. So there's approximately 1,000 recordings of these out in the wild. This is a map from Atlas of Living Australia. They're found in a rough band across the very rough band across the centre of Victoria, from west of the Grampians. So the Grampians are here, and there's this population on the other side, and then all the way across to Latrobe Valley and Yarra Valley in the east, and this random guy out here. So basically they have small buds, fruit and flowers, and have undulating round leaves, and they're really, really similar to Eucalyptus ovata, which is a swamp gum. They actually hybridise as well. One of the distinguishing features though is that it has rough bark all the way to the outer limbs most of the time, not all the time. It also has a cyanic compound in the leaves, which is an anti-herbivory trait. And it's been suggested by previous studies that the cyanic compounds in the leaves of each individual directly relates to the genetic diversity of that individual, which I would like to investigate if I get time. It'll be icing on the cake, hopefully. Got to get the cake baked first. So basically I'd like to see if there is a correlation between genetic diversity and the cyanics, or whether it's environmental factors that are impacting the level of cyanics. Or I was just listening to some of the talks yesterday about somatic mutations, and that's really interesting. So my second species is Eucalyptus strzelecki or strzelecki gum. And so on that map, even though it's a smaller cluster, there's approximately 2,000 individuals. Those are really rough numbers, so please be gentle with me. It's found primarily in eastern Victoria, around the Strzelecki Ranges, which is here. And the biggest population occurs between Warragul and Traralgon, which is this blob here. So when a lot of the power plants and mines went up, a lot of the Strzelecki's were taken out of the population, or, well, removed. They were chopped down. So both species are listed as vulnerable under the EPBC Act, but Strzelecki is listed as threatened under the Victorian Flora and Fauna Guarantee Act, and even though it's listed, it was still removed for mining and power plants and things like that. It doesn't have a cyanic compound, and it's distinguished by these glabrous coating on the juvenile leaves, and a smoother pattern bark which turns pinkish peach in places seasonally, and it's really pretty. It's related to yarragum. They're both kind of swamp gums, and they come under that swamp gum umbrella. So the reason why I chose these two was because yarragum in particular is really getting a big push for conservation now, especially down in the Otways. or down, not in the Otways, but in that region, so around Anglesey and Apollo Bay and places like that. And in fact, there's a great nursery down there and he's really, really passionate about his yarra gums, and so he's doing some great work. And with the Strez, that's in the Yarra Valley and in Churchill as well. Oh, that's not correct. Cool. So there. So there's going to be hopefully some replacement of Strzeleckies when they rehabilitate those mining areas and power plants. So basically, I'm mapping the contemporary distributions of yarra gum and Strzelecki gum, and while I do that, I'm looking at some of the physical habitat that occurs there. So I've been looking at flowering times. These are all just quick notes that I jot down on my field sheet. So crown density, crown health, things like that. Soil type, elevation, those are all being collected for the samples that I am taking genetically. So actually that's not with the mapping. So mapping is separate to my samples. I got into the field much sooner with my mapping because it didn't require a permit and I had some hold-ups with my permit. And so as a result today, I'm presenting like 50 samples. So please don't think that's the be-all and end-all. It is not at all. So I'd like to also determine the population structure of yarra gum and Strzelecki gum using genetic analysis. And like I said before, 50 samples is in no way going to cover that. So please continue. If you see all these things, just continue thinking it is just for now. I will be expanding on that a lot. And developing a suite of genetic markers for each of those species to compare the genetic makeup. And I'll also be looking at current commercial seed stock that's already there to determine if it's representative of what's in the landscape and to inform management. And also potentially, again, if I get time, the potential functional ecology that comes with it. So as Margaret mentioned earlier, insect pollinators aren't necessarily affected detrimentally by fragmentation. When you open up areas in the landscape, it makes it easier for introduced pollinators like your honeybee that can travel 5 k's, whereas your native bees, a lot of them only travel a couple 100 metres. So these honeybees are actually more able to move throughout the landscape. So potentially, if I get time and money and all those things, it's another thing to look at. Oh gosh. Okay, so again, these maps are from, I use Atlas of Living Australia to just separate them into general populations that I then sampled from. Again, sampling is ongoing. And the isolates as well, they're really important to this. So again, how many samples I take from each population will be as many as I can, but it depends on the size of the population as well, just to give an idea about the allelic diversity. So the population number 5, this one down here, was investigated by Linda Broadhurst and the Landcare groups down there and found some pretty cool stuff, not as isolated as we thought it might be. And that's individuals within that population. So I'd like to do the same thing but expand it to all the populations. So again, same for Strzeleckies. Areas of re-veg to put back in what was taken out for mining and logging. Hopefully, well, that's the wrong button again. In this area and, well, wherever it can go in properly. So I'm new to genetics. This is my very first genetic project, so please be gentle with me. I'm using Kaizen Power Plant Pro Kits, so I thought I would talk to our last speaker about that because I've been getting some stuff out of it. And I'll be using SNPs for DART sequencing. So I don't have any analysis yet, but I thought I'd present my preliminary results. So mapping these distributions to define habitat preferences has been going really well. We've found that yarra gum, this is just yarra gums by the way, I've only just started Strezzies, We found that yarra gums really prefer the plains grassy woodland herb-rich foothill forests EVCs, and they really prefer sandy loam, and loam sand soils, which are their favourite. And these are the exact same areas which have most commonly been used for agriculture, urbanisation and mining. So I've already used kind of these parameters to find a few new populations in the landscape that weren't previously recorded. And hopefully this information can help with future re-veg efforts as well. So the orange in this map shows modified pastures, which could be potential re-veg areas, but this is distribution range of yarra gums from Flora Victoria, and you can see how well they overlap. That's pretty much 1 for 1. So this map is really in its infancy. It only has 400 points on it so far. This is yarra gums again, and I noticed this morning that my GIS data didn't overlap there, so where there should be, there's a white patch there, there should also be one here. There's not, I'll have to fix that up. So basically this is a heat map, but it's not really a heat map. It's just showing the higher frequencies of individuals that are closer together. So that is, again, ongoing and continuing. So basically at the moment we're still gaining an understanding of the population genetic structure of both species. Again, this is just yarras so far. So the connectivity is really, and the gene flow is hopefully what we're looking at. The genetic analysis of our samples will give an indication of the connectivity of these populations throughout the landscape, as well as between the individuals within the subpopulations. And hopefully, again, if we get time, it'll be a conduit for the pollination regimes. It'll indicate possible barriers or if there's no barriers, and we can do that with landscape genetic techniques. So I've popped up age classes there. So far, observations and recordings suggest that there's plenty of adult trees that are younger than the age of European fragmentation, and that suggests that their genetic makeup may be influenced by fragmentation through isolation. So we were originally worried that if all the yarras in the landscape were remnants, we'd have nothing to test because they were all conceived before European fragmentation. But yarra gums, luckily, are considered medium to fast growers, so I have plenty of adult trees. There's not as many recruitment or young, but there is still some that we can test to see if isolation is an issue. So, final outcomes. We'll be analysing the genetic makeup of seeds, what's in the current seed stock in commercial areas. Oh, I'm so close. We're talking to the local Ballarat seed bank, and a lot of the yarra gum seeds there coming up are actually hybrids, so we need to have a look at what's going on there. And hopefully this can inform land care groups, catchment management authorities, local government, places like that, for selecting the correct seeds for future resilience. That's it.